Hello, and welcome to Gairdner Science Week 2021. I'm Janet Rossand, and I'm President and Scientific Director of the Gairdner Foundation. I hope you've been enjoying the events of the week so far, and still more to come. Before we begin, I'd like to acknowledge that the Gairdner Foundation is located in Toronto, Canada. And we acknowledge that we're on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, the Wendat peoples, and today Toronto is still home to many diverse First Nations, Inuit, and Métis people. Today's Canada Gairdner International Symposium is From Gene to Genomics and Epigenomics, the Future of Cancer Precision Medicine. You'll hear from 2021 Canada Gairdner Award laureates Mary Claire King and Elizabeth Eisenhower, as well as international research leaders from Canada, the US, and the UK. Today's events wouldn't be possible without our sponsors, so I'd like to particularly thank our Gold Gairdner Week sponsors, the University Health Network, York University, the University of Toronto, Sinai Health, Queen's University, London Drugs, TELUS Health, and McGill University. I'd also like to thank our partners, the governments of Canada, Alberta, and Quebec, CIHR, and our media partner, the Globe and Mail. You can learn more about all of our sponsors by visiting the Expo booth section located on the left-hand side of your screen. Our speakers will be answering questions at the end of the symposium, so please be sure to put your questions as we go along in the chat box on the right-hand side at any time as we go through the speakers. And now I'd like to introduce our chair for the first half of today's symposium, Dr. Jim Woodgate. He's a long-time supporter of Gairdner and is now the President and Scientific Director of the Terry Fox Research Institute. Thank you, Jim, for joining us.
Thanks, Janet. It's my great pleasure to co-chair this symposium on behalf of the Terry Fox Research Institute. And indeed, you're right, I've had a long relationship with the Gairdner Foundation, and I personally want to extend my congratulations to all of this year's laureates, and to you, Janet, and the Gairdner team for a remarkable week of scientific exposition. As always, it's been fantastic. So our first speaker this afternoon is Dr. Mary Claire King, who is the 2021 Canada Gairdner International Award laureate from the University of Washington in Seattle. And her lecture is called How Heritable Cancer Mutations Inform All Cancer Treatments. Over to you, Dr. King.
Good morning and afternoon to everyone. It's a great pleasure to be with you again today. This has been an amazing week. Janet, you are beyond belief energetic, positive, and on top of all of the details. I am learning a tremendous amount. What I'm in fact going to talk about today are strategies that we are using to identify cryptic inherited mutations in families that are severely affected with breast or ovarian cancer, but who have no mutation identified in BRCA1 or BRCA2 or any of the other homologous recombination repair genes. I picked this topic both because, of course, it's enormously important to me and I think important clinically, but also because the types of lesions that we hope to identify will be meaningful, I expect, not only for inherited predisposition, which is my interest, but also for the classes of lesions that all of the others of you are already identifying in tumors. So on this first slide, we see just a few of the families in our series of now something over 6,200 families who share the feature that there are multiple women with breast or ovarian cancer. And for the women on this slide and many others, we do not know the genetic cause of that predisposition. To try to get a sense of how big a problem this was, and it's difficult to know because the families that come into our studies are, of course, invariably those who have not had positive results by conventional genetic testing. And that, of course, has been goalposts that have moved over the last decades. But just to get a sense of the size of the problem, years ago, when the patent was first abrogated and testing became very, very widespread, I evaluated just under 1,000 families in our series who had at that time no mutations identified in BRCA1 or BRCA2, either by conventional genetic testing, that is, no coding sequence mutation and no nearby flanking mutations. And in those roughly 1,000 families, we solved 20% by mutations in all the other homologous recombination repair genes, but 80% were left unsolved. That's obviously not 80% of all breast cancer families. Many, many, many are solved by conventional mutations, by good genetic testing, but it's not a small problem. There are many of these families out there. It's the view of those of us working in the field, and Will Folks and I had a chance to chat about this yesterday, that the critical genes have been found. The genes that harbor mutations that lead to very high risks of breast and ovarian cancer have been identified. There's about 10 of them. I refer you to an excellent paper by Will on this topic. But what has not been clarified or not been known yet is the very large number of mutations of non-conventional sorts that can occur in these genes. And I'm thinking particularly of the fact that tumor suppressor genes, by their very nature of being tumor suppressor genes, by the nature of being genes in which somatic mutations occur quite frequently, have very, what I call, messy non-coding regions. And these can harbor complex structural genomic mutations that are not detectable even by good conventional genetic testing. And then of particular interest to us, and something that I'll speak a few minutes about at the end of my talk, but very much still work in progress, is the possibility of distant regulatory regions for these genes, which can, of course, apply to any gene, some of which have tissue-specific effects, that is, some of which alter gene expression, but not in a way that is detectable in blood. So let me plunge in. First, just to get us warmed up, let me illustrate the principle of using whole genome sequencing, in this case short read Illumina whole genome sequencing, to identify lesions in this case involving BRCA1 that would not have been identified by conventional genomic genetic testing. And in this case, the young woman with breast cancer is a young woman of Mexican ancestry. She was diagnosed with triple-negative breast cancer at age 35. Her mother had died of breast cancer. And as you see, her DNA from her blood, when subjected to short-read whole genome sequencing, revealed a 240 kb deletion that included BRCA1 and 5 other genes. A couple of interesting points were that she had no systemic effects. She had obviously very high risk of breast cancer. She underwent a subintuophorectomy, so she did not develop ovarian cancer. But clearly, haploinsufficiency for the other genes did not have any impact on her. Interestingly, this mutation turned out to be a founder mutation in Mexico, in Native American population in Mexico, and has proven to be very useful because we were able to develop diagnostic primers as soon as we had the breakpoints. And in clinics in Mexico that don't have the capacity to carry out complete genomic sequencing, it is nonetheless perfectly straightforward to do just PCR with diagnostic primers for this mutation in communities where it is quite common. So that's the warm-up. A few years ago, Sylvia Cassidy, Suleiman Gulziner, and others in my lab developed a use of our multi-gene testing platform, BROCA, which Tom Walsh had developed about a decade ago, specifically for looking at transcriptional alterations. And we call it cBROCA for cDNA BROCA. And by carrying out analysis of cDNA, still now entirely from lymphoblasts or blood of our participants, we're able to identify mutations that do not appear at first blush with genomic sequencing to be important, but when we look at their transcriptional effects, we can see from the sashimi plots that there are effects. And cBROCA has the advantage that we can do this simultaneously across all the genes that we target, which are all of the homologous recombination repair genes, so we don't have to know in advance which gene might be critical. So what I've done on this slide is put several examples of interesting mutations in BRCA1, these first 4 families, that are in fact exonic, that is, they're within exons at the genomic level, but they alter either splicing enhancers or splicing silencers, and consequently have transcriptional effects as shown on the sashimi plots here. So the red sashimi plots indicate the families in question, and the blue sashimi plot is from a control. Similarly, for BRCA2, the same sort of thing applies. Exonic mutations appear and can alter but can have transcriptional effects because they are in fact mutations in enhancers or silencers, and they're not relevant at the level of protein changes. Many of them are in fact silent at the level of protein change. Those of us who are devoted to this field know that there are certain suspicious exons in BRCA1 and BRCA2 that are particularly prone to these kinds of events. But because, of course, all of the mutations in BRCA1 and BRCA2, indeed in all of the homologous recombination tumor suppressor genes, are individually rare, new ones of these pop up all the time. So being able to sequence with CBRCA, to sequence transcripts, it enables one to reveal these in a systematic way without having to know in advance at which gene to look. At the bottom here, I've put a particularly interesting lesion. This one happens to be an APC, so it's in a colon cancer family, not a breast cancer family. But this lesion is deeply intronic, as you see here, and has led to the creation of a pseudoexon in intron 5 of APC and an internal stop as a consequence of that pseudoexonification. So CBRCA is a useful tool for carrying out this kind of analysis, and this we published a couple of years ago. 2 much more recent approaches that we've used are ones that I'd like to speak a little bit about here. The first is called CATCH, which we pair with multiplex long read sequencing, and it works like this. So this is a scaled down version of one of the families that was on that first slide, just a reduced version so it would fit. So complex mutations in repetitive non-coding regions of DNA are not detectable by short read sequencing. So some are, but not all. So what we did in the case of this family was combine whole genome PacBio long read sequencing, which is extremely expensive, as we all, of course, know, with a method of enriching for all of the target genes that we care about, and then multiplexing multiple samples. So my former postdoc, Han-Li Ji, who is now on the faculty at Stanford, developed a method called CATCH, which allows one, it's kind of like old-fashioned pulse field Joe electrophoresis, except it's CRISPR-Cas assisted. It's Cas9 assisted targeting of chromosomal regions, and it allows us to target a couple of hundred kb around each tumor suppressor gene, so entire loci. Then to use CRISPR excision of those fragments from live cells, in our case from lymphoblast lines of our patients. And that excision yields long, intact DNA fragments that are very good for long-read sequencing. And here's how that works. There's a device that, as I said, looks like a modern pulse field gel electrophoresis device that allows us to take freshly grown patient lymphoblasts, about 750,000 cells per patient, lyse the cells to release the intact DNA, add the CRISPR guides and apply a charge to the DNA, add Cas9, then incubate, then separate the cut fragments that are separated by the charge by size, elute those fragments, and collect based on size. This enriches for, in a quite nice way, each of the targeted loci. Here's an example of the enrichment for BRCA1. Then for all of the targeted loci, and we've been targeting 6 or 7 of the critical breast and ovarian cancer genes in these experiments, for the targeted loci, from multiple individuals, we can now multiplex PacBio sequence so that we need buy only one flow cell for multiple tests. We select the roughly 10 kb reads so that we have very good coverage from each targeted locus. And in this example of the family that I showed on the previous slide, what we saw was that in BRCA1, intron 13, there was an insertion of about 2,800 base pairs that was revealed by these soft clipped reads on either side. And when we looked at the sequence carefully, here's what it looked like. The insertion, which was not detectable by short read whole genome sequence, it was not detectable by any of the other tools to which we had turned in trying to solve this family. The insertion is 2,856 base pairs, and it introduces an SVA, that is a retrotransposon that's the combination of a SINE, a VNTR, and an ALU element. That retrotransposon creates a pseudoexon, which includes an internal stop. And here's the structure of what it looks like. And the internal stop is embedded in it when it reads in frame out of exon 13 of BRCA1 and into this intron. And at the level of transcript, we can see that several transcripts are made in the lymphoblast lines of the patients in this family, including both the SVA at the genomic level and then two different versions of pseudoexons at the transcript level, both of which include internal stops. We made diagnostic primers for this event, and as you would expect, it co-segregates with breast cancer in the family. This class of events, and this particular event is from a family, an American family originally from Romania. So I'm hoping to be able to work with colleagues in Romania and see if there are other families with the event. This particular event does not occur in any of our other families, but this class of events certainly could obtain. And finally, an approach we're working on now, so this is very much a work in progress, is the following. We're trying to use high-C chromatin capture to identify regulatory regions that may be tissue-specific. And this approach is motivated by the family that I show here. This is our family 923. So family 923 is severely affected with breast and ovarian cancer, as you see here. Indeed, one child developed and died from Wilms tumor, although that may have nothing to do with the rest of our story, I don't know. But it has no responsible mutation identifiable by anything I've told you so far, not by short-read whole genome sequence, not by exome sequencing, not by C-BRCA, nothing. So in other words, there is no transcriptional difference between a hypothetical mutant allele compared to wild-type alleles at any of the known homologous recombination repair genes. So we said to ourselves, what on earth is going on with this family? We carried out low coverage PacBio sequencing, just 8x average. Obviously, that means that many reads are far less than, or many sites are far less than 8x coverage, and discovered a 34 megabase inversion on chromosome 17. So one end of the inversion is just at the it's just at the edge of the centromere on the P arm, and we know this thanks to the telomere to telomere sequence. The exact locale of this end of the inversion was not detectable until T2T came online, and the other end of the inversion is in the midst of chromosome 17. So another chromosome 17 event. As many of you know, several of the critical genes for inherited breast and ovarian cancer are on chromosome 17. 3 of them, RAD51D, BRCA1, and RAD51C, are within this inversion, so they are inverted in the patients in the family who carry the inversion. And P53 is proximal to it, and BRIP1 is about a megabase distal to it. So we have an inversion, which is detectable again by observing the soft clipped reads from the PacBio sequence. The inversion does not disrupt any annotated gene at either end, let alone any gene critical to breast or ovarian cancer. It's in a gene desert. The inversion breakpoint is in a gene desert in the middle of 17 17q and is at the edge of the centromere in 17p. We made diagnostic primers for the inversion, and as you can see, it co-segregates with breast cancer in the family, as shown here. So we now have a conundrum. What might this inversion be doing that is not detectable at the level of transcripts from blood or transcripts from lymphoblasts? Because recall that although, of course, we can detect the inversion in blood and lymphoblasts, it's a genomic event, there is no imbalance in transcription in blood or lymphoblasts in transcripts from any of the people who carry the inversion. So what we decided to do was to undertake high-C chromatin capture. And the idea here will be to try to identify regulatory regions for which one end of the regulatory event is in a promoter or regulatory site of a critical gene on chromosome 17, and the other end is on the other side of the inversion, so distant events. Just to review very quickly, recall that high-C chromatin capture involves cross-linking DNA in the nucleus of a cell, then, as shown here with formaldehyde, then cutting with a restriction enzyme, filling the ends and marking them with biotin, ligating the ends, then purifying and shearing the DNA, pulling down the ligated ends with biotin so that one now has the combination of the two ends of the two regions that are quite distant in two-dimensional space, but are now paired in three-dimensional space, and one can determine by straightforward sequencing what they are. So our first experiment was to compare results of high-C chromatin treatment for women from the family who carried the inversion versus those who did not. Now, we don't expect to see in their lymphoblasts genomic DNA, we don't expect to see the critical events we're looking for because, of course, we expect these to be tissue-specific events. But we do hope that high-C can detect the inversion. So just to reiterate that, given that the inversion does not alter expression of breast cancer genes from lymphoblasts, we don't expect the lymphoblasts to reveal the critical long-distance interactions. That will require data from relevant tissue. But the first experiment is just to test a high-C and see if we can get it to work. And we can. Here is just a couple reduced heat maps to illustrate the point. So from a person from the family who has wild-type sequence, doesn't have the inversion, we have a simple heat map. I've just excerpted the critical bits of it around each of the homologous recombination repair genes. These are the breast cancer genes on chromosome 17. If we compare that to the number of reads indicating events in a person with the inversion, it looks just the same around each of the genes as we expected. We don't see any transcriptional effect in lymphoblasts, but there's good evidence for new chromatin relationships at the site of the inversion itself, both in 17p and in 17q. So this is just a proof of principle. This is just to convince ourselves that we can detect the inversion itself with high-C. So what will we do next? We're going to undertake high-C on HEMEC cells in order to try to identify regions that we care about. And we will look at these same genes. We'll focus on chromosome 17. We'll focus on all of these genes on chromosome 17 to see if we can find regions that are revealed by high-C that are specific to breast epithelial cells that are distant regulators. And to do that, we're going to target the TADs, the transcriptional activation domains of all of these chromosome 17 genes, so as to focus our coverage and increase our efficiency. We call this high-C BRCA. It's another way of targeting just the regions we want so that we can increase with at reasonable cost our coverage. Then what we'll do from a carrier from our family who carries the inversion, we'll see if we see any interesting effects in that person. Now, a really pie-in-the-sky experiment that we hope to be able to do is from a carrier of the inversion to take her blood sample. The family is very involved with us and they're happy to give us blood and so on, and see if we can de-differentiate her blood cells into embryonic stem cells, and then carry out the same experiment in the the de-differentiated blood cells, the embryonic stem cells of a person with the inversion from the family. So while the motivation of this approach is to try to solve this one family, obviously the goal is to use this approach to identify distant candidate regulatory regions in families who don't have any inversions, in families generally who are not solved, and to go back to the many families who are not solved and sequence their now revealed distant regulatory regions that were revealed in HEMEX, to see if there are any point mutations in those regions that might have effects on the genes that hypothetically are regulated by those distant genomic sites. And to do that, of course, we will carry out individual assays. So that's what's up next in our lab. We'll see if we can wrangle high-C chromatin capture to our service for identifying these kinds of events. So everything that I've told you illustrates our theory of mutation, which is that anything that can go wrong will. But the rather heartening realization that mutations have a physical reality, and if they are out there, we should be able to find them, and we should be able to characterize their effects. Thanks very much. These are the young people of my lab who carry out the work that I've described, and I look forward to talking with you about this approach, and I look forward very much to hearing the next speakers. Thanks so much.
So thank you very much, Dr. King. Actually, nothing went wrong with that presentation for sure. Thank you. So up next, we have Dr. Charles Swanton. He's Cancer Research UK's chief clinician, and he's at the Francis Crick Institute in London. And his lecture is called Tumor Heterogeneity and Tumor Evolution. Over to you, Charles.
To Dr. King and Dr. Eisenhower. It's a great pleasure to be here today, virtually. So I'm going to talk about tumor heterogeneity and evolution and the role of chromosomal instability in immune evasion. Here are my disclosures. So we set up TRACER-X, a study to monitor and track cancer evolution over time and space about 7 years ago, with the help of these talented scientists on the right-hand side, clinician scientist Dr. Jamal Hanjani, and two scientists in the group, Dr. Rowan and Christina Lombardelli. And the aim of TRACER-X is to track 842 patients with early-stage non-small cell lung cancer from diagnosis through to either cure or death. And so when I say track, what I mean by that is that we subject their tumors to multi-region sequencing to reconstruct phylogenetic trees of those tumors to infer the trunk mutations that are present in every tumor cell and distinguish them from the branch mutations present in some cells but not others, to understand what the drivers of diversity and ultimately clonal expansions that may lead to metastatic disease are, from diagnosis through to either cure or death. And the premise of the study is that deep spatial tumor sequencing will help us distinguish metastatic competent from incompetent subclones within the same tumor. So I want to just go through now some of the results from this study over the last 5 years or so, and where we are and where we're heading, I hope. So first of all, I want to talk about what are the sort of underlying basis of clonal mutations, that is truncal mutations in the tumors of these lung cancers, early stage lung cancers that we're sequencing. And there are three major types of mutational processes in these early stage lung cancers that we see: a tobacco carcinogen signature, and then 2C to T mutation signatures, the Apobex cytidine deaminase mutation signature, and spontaneous deamination, so-called mitotic clock aging signatures. When we look at the clonal mutations, these are dominated by C to A mutations derived from the smoking signature, signature 4. These dominate the trunks of the tumor's evolution trees. Indeed, in lung adenocarcinoma, the number of cigarettes that patients have been exposed to correlate with a number of C2A mutations in the trunks of the tumor's evolutionary trees. Sukit Chu in the lab, in collaboration with Monty Winslow in Stanford, has been using an elegant approach that Monty derived called TuberSeq to monitor the evolution of tumors in autochthonous mouse models in non-small cell lung cancer and adenocarcinoma KRAS-driven, p53-driven mutant non-small cell lung cancer. And in this work, he's managed to establish, with Monty's support, 3 real types of tumor suppressor genes that we're beginning to see. So we're seeing tumor suppressor genes that are inactivated in early stage non-small cell cancer, many with these sort of C2A mutations that we see in the trunks of these tumors that drive tumor initiation. And then others don't drive tumor initiation, but actually drive expansion of previously initiated tumors. So they are very large tumors in these mouse models. And then you've got some that drive very rare but exceptionally large tumors. We call these the hopeful monsters, typified by suppression or silencing of DNMT3A and CDKN2A. So what about mechanisms of branched evolution? Well, here we see a different mutational process. These are the mutations that characterize cell-to-cell variation in these early stage lung cancers, many of whom are current smokers. Here, the mutational signatures are dominated by the APOBEC and mitotic clock C to T mutations. So even in a patient who's smoking maybe 20 to 30 cigarettes a day, it'll be the mitotic clock signatures and the APOBEC signature which will be dominating cell-to-cell variation at presentation with early stage lung cancer. So this prompted us to study the role of APOBEC in the context of adaptation in early stage lung cancer. And this is work from 4 talented scientists in the group: Nana Kanu, Meni Venkatesan, Debbie Caswell, and Mihaela Angelova. And I just briefly review this published work that came out earlier this year, and unpublished work that's currently on bioarchives under review. And this is work from Debbie Caswell, where she's created an APOBEC3B mouse model where she can induce the human transgene later on in tumor evolution after EGFR L858R induction to address what is the impact of later or subclonal APOBEC3B induction, mimicking what we see in human tumors following EGFR tyrosine kinase inhibitor exposure. And what we see is that tumors that are expressing APOBEC3B develop more rapid resistance to the tyrosine kinase inhibitor erlotinib in this mouse model compared to the EGFR L858R mice alone. And Mihaela and Mani and Nena in the lab showed that APOBEC3B silencing in type 2 pneumocytes could suppress chromosomal instability and the proportion of cells with micronuclei in type 2 pneumocytes. And following expression of APOBEC3B and HEC293 cells, they showed that you could increase the proportion of cells with anaphase chromosome segregation errors and the proportion of cells with micronuclei, indicating that APOBEC3B not only drives cell-to-cell variation in the context of C to T mutations, but may also drive karyotypic variation of chromosome number and possibly chromosome structure. So then what about the clinical endpoints of TRACERx, which was trying to establish what the impact of heterogeneity, genetic heterogeneity, may be in the context of somatic evolution of these tumors, clinical outcome. And interestingly, we did not find a relationship between point mutational diversity and clinical outcome, but what we did find was that tumors with high levels of chromosomal instability were associated with poor clinical outcome. So really, this is the point of my talk now, is what is the role of chromosomal instability in tumor evolution over space and time? And firstly, does chromosomal instability fuel ongoing selection? And this is work from Amelia and Tom, co-supervised by a fabulous PI in the Lung Centre, Nikki McGranahan, a previous PhD student of my group. Now, we wanted to address whether chromosomal instability would stimulate neutral evolution or perhaps positive selection. And this is work that we published about 12 months ago now. We found evidence that chromosomal instability later on in tumor evolution could fuel diversity and ultimately parallel evolution of copy number events that were present in about 1/3 of tumors, where you would get, for example, amplification of MYC from the maternal alleles in one subclone of the tumor and from the paternal allele in another subclone of the same tumor. We find ordering of these copy number events over space and time, with tumor suppressor genes lost early, as you might expect, and then amplifications of many oncogenes occurring intermediate or later on in tumor evolution, with one exception, HLA loss of heterozygosity, that commonly occurs as a late event in tumor evolution. And then, in keeping with selection, we find that mathematical models are consistent with a selection process over both space and time, both early on in tumor evolution and later on in tumor evolution, driven by chromosomal instability. We also find evidence in non-small cell lung cancer of genome doubling events being very frequent. About 75% of our tumors show evidence of genome doubling. And when we see it, it's almost always a clonal event, almost always a truncal event in tumor evolution. And when we modeled this about 4 or 5 years ago, as Dewhurst in the group, who recently, well, actually some time ago, moved to the Rockefeller with Tisha DeLange, showed that the genome doubled HCT116 clones become very much more chromosomal unstable over 18 months in culture, shown in red here, relative to their diploid progenitors or parental cells, shown in blue. So it seems that by becoming genome doubled, cells can explore the copy number space much more rapidly and become chromosomally diverse much more rapidly in culture, suggesting that genome doubling, along with APOBEC, may also be an ongoing driver of karyotypic diversity and chromosomal instability. So for the last sort of 10 minutes or so, I want to talk to you about how chromosomal instability may impact immune evasion. And this is again work from TracerX, where we sampled multiple regions and asked what the impact, or what are the cell intrinsic mechanisms of immune evasion in these tumors, in these early stage, untreated non-small cell lung cancers. And these are the 3 mechanisms I'm going to focus on for the next 10 minutes or so: HLA loss of heterozygosity, neoantigen evolution, and antigen presentation disruption. Nikki and Rachel created this tool back in 2017 where you could infer HLA class 1 loss of heterozygosity from whole exome sequencing data. And in this study, they found that HLA-OH is actually extremely common. About 40% nearly of tumors, early stage lung cancers in TracerX, have loss of 1 or more class 1 HLA allele. When we see this, it's almost always subclonal in nature. Well, not almost always, but it's enriched for being subclonal relative to a clonal event, which was unexpected. So here are some of the minority of tumors where we see clonal HLA copy number loss of class 1 alleles. And here's some examples of subclonal HLA class 1 copy number loss. I'm just going to zoom in on 4 particularly interesting tumors shown at the bottom here, which have got parallel evolution for loss of the same HLA haplotypes in 2 separate subclones from the same tumor. And 2 of these tumors have got clonal genome doubling events. And so what this means is that for them to lose for them to lose 1 HLA haplotype in 2 separate subclones, they have to have lost the same class 1 allele on 4 occasions because of this clonal genome doubling event. And consistent with selection, you can see the 6B loss of heterozygosity events occurring in squamous cell carcinoma as being probably the most potently selected events across the genome occurring subclonally during tumor evolution. So what impact does this have on emerging subclones? Well, what we find is if we look at clones that have lost, subclones that have lost HLA relative to adjacent subclones in the same tumor from the same patient that haven't lost HLA, we find that the subclones that have lost HLA tend to have significantly more mutations in them than the subclones that haven't lost HLA. And when we ask where does HLA-LOH occur in tumors, we tend to find HLA-LOH occurring in immune hot tumors with a brisk lymphoid infiltrate. We also find evidence for a trend, at least. This wasn't significant from this small cohort, but we're seeing a trend towards enrichment of HLA loss of heterozygosity at the transition from the primary lung cancer tumor to the central nervous brain metastasis. And we're expanding this analysis from Brassianos and colleagues in a much larger cohort of about 200 primary metastatic tumors from TracerX, work in progress. So what else is happening in immune hot tumors? Well, we also find evidence for antigen presentation disruption. So we find evidence for disruption of the antigen presentation machinery, either through copy number loss events or through deleterious point mutations, that appears very often to be mutually exclusive with HLA-LOH and enriched in immune hot tumors. So what are we going to do about this sort of inexorable diversity clinically? So we've been thinking for some time that what we need to do is target multiple clonal mutations to essentially render drug resistance next to impossible. But to do this, we have to target multiple unique clonal mutations because no two tumors will have the same panoply of clonal mutations. So we've been thinking about this in quite some depth, and what we noticed about 5 years ago was that smokers or ex-smokers tend to have very many more clonal mutations in their genomes than never smokers, suggesting that perhaps these clonal mutations, of which they've got about 5-fold more, might be subject to immune surveillance and might be clonal neoantigens and might be tumor vulnerabilities that we could exploit for patient benefit. So the question is, are these trunk or clonal mutations important? And Kevin Litchfield and Nikki McGranahan and others in the lab have been looking at this to ask whether clonal mutational burden or clonal neoantigens might be important for checkpoint inhibitor therapy benefit. We got an early insight into this back in 2016, where Nikki and Rachel found that anti-PD-1 responders had more homogeneous tumors and a higher clonal neoantigen burden. So they had more neoantigens in the trunks of the tumor's evolutionary trees. And this was work that was subsequently validated and substantiated by Ellie Van Allen and colleagues in a much larger cohort of 244 microsatellite stable tumors. Recently, Kevin Litchfield in the group and James Redding and others in the group have shown through an analysis, a meta-analysis of 1,000 patients treated with checkpoint inhibitors from across the globe in different studies shown here at the top here, that if you break tumor mutational burden, which is known to be a biomarker of checkpoint inhibitor response, into its constitutive components, that is trunk and branch tumor mutational burden, it's the trunk mutations that best predict response to checkpoint inhibitor blockade rather than branch mutations. So truncal mutations seem to predict response to checkpoint inhibitor therapy. So the question we've been trying to ask is, are branch mutations just not noticed by the immune system, or are they actually bad? Does diversity fuel immune evasion? And I just want to briefly highlight work from Gairdner Samuels at the Weizmann Institute that we were fortunate enough to collaborate on, where she created tumors in syngeneic immune competent mouse models with a B2905 mouse melanoma model, which she exposed to UV and created single cell clones. And then she could artificially manipulate the degree of heterogeneity in the tumor she transplanted back into the mice. And what she showed from this work that was published in Cell in 2019 is you get poorer immune control if you have more diverse and more heterogeneous tumors. And both the number of clones and their genetic diversity plays an important role in mediating tumor growth and rejection. And interestingly, the more heterogeneous tumors were associated with a more immune suppressive environment. So with that in mind now, and this is where one of my conflicts of interest come in, so please bear this in mind, we're now leveraging clonal mutations to identify T cells in tumors that specifically recognize clonal mutations. And we're expanding those and we're giving them back to patients now in the context of phase 1 trials shown here. And bear in mind, this is where one of my conflicts of interest come in. Now, just before I finish, I just want to quickly highlight really this ongoing tug of war and the predator-prey relationship in tumors between the immune system and the evolving tumor. And really this speaks to how immune evasion occurs in immune cold tumors, that is tumors without an immune infiltrate. And what we find in these tumors is that they have evidence of loss of clonal neoantigens. So presumably truncal neoantigens that once were immunogenic, but due to evolution and selection of subclones that lack the clonal neoantigens that we specifically see here on the right-hand side in immune low tumors or tumors that don't have a brisk lymphoid infiltrate. This perhaps is a fossil record or an indication of prior immune reactivity and selection of subclones that have lost clonal neoantigens through chromosomal instability. So I've shown you very briefly 3 mechanisms through which chromosomal instability we think can fuel immune evasion and selection in the context of a predatory immune environment. And tumors that don't show evidence of these mechanisms are associated with a better clinical outcome in TRACERx. And lastly, I just want to quickly say that we're focusing on ctDNA hallmarks of recurrent disease in the early adjuvant setting now, using minimal residual disease tools that we're collaborating with Archer Diagnostics and others to create bespoke panels where after surgery we can use ctDNA to detect early recurrence, before heterogeneity has set in, we hope, before there's evidence of macroscopic disease on a CT scan, where we get the indication of these truncal and branch mutations occurring prior to macroscopic relapse on a CT scan, up to more recently 141 days, a median time, minimal residual disease times now. So we hope to be able to intervene, and with 2 trials we're running with AstraZeneca and others, we hope to be able to see evidence where we can intervene off a patient's checkpoint inhibitor blockade and eradicate emergent residual disease before there's evidence of macroscopic disease on a CT scan. And early evidence from Tom Poles and colleagues in collaboration with Roche showed that atezolizumab, anti-PD-1 therapy, is specifically beneficial in the ctDNA positive cohort using the MRD biomarker tools that were created from TracerX and subsequently given FDA breakthrough approval. So I've hopefully given you some indication of the role of chromosomal instability in tumor evolution, how APOBEC and genome doubling may fuel chromosomal instability as well as genome doubling, and its impact on the immune microenvironment and ultimately selection for immune competent subclones that can ultimately evade the predatory immune system. So I'd just like to thank all the people that made this work possible, including CRUK, BCRF, the ERC and the Royal Society, all of the lab, and of course the TracerX team, and most importantly patients and their families. Thank you very much for listening.
Thank you very much, Dr. Swanton. Great lecture as always. So next, moving on, I'd like to introduce Dr. Sam Aparicio from the University of British Columbia and the BC Cancer Research Institute in Vancouver. And his lecture is entitled Molecular Taxonomy of Breast Cancer and Next Generation Treatments. Over to you, Sam.
I'd like to address 2 topics today. The first is to address some work, just review briefly some work we've been doing over the last 10 to 15 years to understand the variation between breast cancer patients among individuals. And this pertains to questions around identifying how patients have distinct cancers and how to manage and treat them. And the second is revolving around why cancers vary within individuals and what we can do to understand the clonal evolution that's taking place and its relationship to drug resistance. The background to breast cancer can be probably summarized on the variation between patients on this slide sort of illustrates how over the last 20 years, our understanding of breast cancer as a single label, in fact, covers up many diseases, because the variations between patients have now been understood both from transcriptomics, which first started to make its way in over the sort of clinical standard, where breast cancers are considered to be initially divided up by expression of hormone receptors and amplification of the HER2 gene, both of which pattern therapies. And then with the advent of transcriptomics, 20 years ago, the recognition of expression subtypes, 10 years ago, the relationship between DNA and expression became important. And now with next generation sequencing and other multi-mode genomics methodologies, we recognize that breast cancer may be 10 or more discrete molecular subtypes, each with a phenotype attached related to driver genes and the presence of different forms of genomic instability. And the other vertex of variation which has been impacted by genomics is the recognition that mutational processes lead to evolution of clones. This is something that we started to look into back in 2009 in breast cancers, using the digital counting ability of next generation sequencing devices to separate out clones within cancer. And there have been many, many studies, too many to really now fit on one slide, representing many different cancers. We've heard some of this today, and I'm sure we'll hear some more. And in the second part of my talk, I'm going to try and address how we can decode clonal variation to understand drug resistance. With respect to breast cancer and genomics, the other thing that NGS has brought us is the realization that drivers of breast cancer occur at all scales of the genome, from SNVs to structural variants to copy number variants. And About 15 years ago now, we started a project called MetaBreak, which grouped together 2,000 patients across Canada and the UK, where we had linked clinical outcomes and access to primary tumor material, and using the genomics of the time and subsequent genomic methodologies as they've observed in a number of studies, we've been able to show that you can resolve breast cancers into these subtypes with distinct outcome differences. And that by combining studies from TCGA and ICGC, Cosmic, MetaBreak together, that we can identify around 40 recurrent drivers of different breast cancer outcome phenotypes, indicating the different biologies which are taking place in the genome. One of the still heterogeneous subgroupings that we've been studying for a number of years are the triple-negative breast cancers, defined by absence of hormone receptors and absence of amplification of the ERBB2 gene. This is still a heterogeneous group. It's probably at least 3 or 4 distinct phenotypes. And one of the realizations we came to early on is that triple-negative breast cancers differ in the degree of genomic instability, as kind of summarized on this diagram here. And in more recent studies, now using single-cell sequencing, we've been able to extend this observation into identifying discrete patterns that are conserved not only in breast cancers, but in high-grade serous ovarian cancers. And what this heat map shows is, from a combination of single-cell sequencing and whole genome sequencing, some work that's currently under review, that we can identify structural patterns, particularly structural copy number patterns, which indicate cancers that are either defective in homologous recombination repair deficiency, and cancers that share fold-back inversion type patterns. And these cancers appear to differ in the degree to which they are resistant to conventional therapies. So fold-back cancers appear to be slower growing and relatively more platinum resistant for reasons that we don't fully understand, in contrast to HRD. But what this underpins is the idea that genomic instability in and of itself is an important phenotype in breast cancer and in ovarian cancer, and something that is pertinent both in terms of biomarker development, but also to exploit therapeutically. And what I'd like to tell you about in the next few minutes is one of the therapeutic avenues that we've been pursuing to exploit genomic instability. And this is based on the idea that in unstable genomes, there are structures that form or can form simply from folding of DNA, which put the genome at risk in an already unstable environment and may sort of kick it over by creating more DNA damage. And the structures that we've been looking at are called G quadruplexes. These form from stable tetrameric interactions of guanine bases in DNA. They're also known to form in RNA. And these structures literally fold up and can act as impediments to polymerases which are involved in replicating the genome, which creates in turn a vulnerability. And it's been speculated that it may be possible to develop small molecule drugs which would, under certain conditions, stabilize these quadruplexes in genomes that are already disabled for repair deficiency, and that would create a therapeutic vulnerability. And indeed, there have been some tool compounds, such as the two shown on the left here, that can produce these effects in cells. But a few years ago, we set out to try and look for molecules that would accentuate homologous recombination repair deficiency, and we're able to identify through repurposing a clinical candidate molecule shown on the left here, which we found was highly active, selectively active in cells that have BRCA1 or BRCA2 deficiency, and producing about a log order of therapeutic index benefit. And we were able to show that this particular molecule, which had previously been thought to be an RNA polymerase 1 inhibitor, actually has G quadruplex binding and probably partial topoisomerase inhibition activity, and that in BRCA-deficient xenografts and BRCA-deficient cell lines, that this produces a therapeutic window. But of course, it's one thing to have the molecule in xenografts and cell lines. Of course, we wanted to go on and use this as a clinical candidate to try and test in patients. And here we've been able to leverage one of the great developments that Elizabeth Eisenhower set up in Canada, or helped set up. The Canadian Clinical Trials Group helped, together with Stand Up to Cancer, perform a phase 1 clinical trial of this molecule, which has recently finished. This phase 1 study was designed initially, of course, just to look at safety and efficacy, but there are, interestingly, some signals of activity. So in the study, which enrolled about 40 patients, there were 4 partial responses and 4 stable diseases, with the longest patient on study being about 15 months. So there were definitely some clinical responses. And intriguingly, from this waterfall plot, what also became apparent is that although the patients enrolled in this study were of mixed genotype, so some had BRCA1 or BRCA2 germline deficiency alleles and others not, nearly all of the responses were in patients that were observed, were in patients with the BRCA genotypes. And an example of that response is shown in the CT on the right, with example of a liver metastasis in a patient that showed an objective response. Even more intriguingly, from post-treatment progression biopsies, so there are 2 examples of reversion mutations. So reversion mutations are of great significance because they tend to imply that either the biomarker or the mechanism are sort of on action, and have been observed with other drug agents that have been used to target genomic instability, PARP inhibitors and platinum. And here in this trial, we were able to observe reversion to reading frame mutations in PALB2 and BRCA2, indicating that likely the biomarker mechanism of action combination is probably in the right ballpark. And so these are very sort of encouraging early signs. There's a lot more clinical development that needs to be done in this area. But what I wanted to just summarize here is that from looking at the genome, we've been able to land on this idea that exploiting genomic instability in itself, using small molecules that directly target the genome, may provide another therapeutic avenue. And this may extend beyond HRD into other forms of genomic instability mediated by theta-mediated end joining and non-homologous end joining in work that's ongoing. The second vertex that I'd just like to switch to, I talked about variation between patients, is now variation within patients. And we heard a little bit about this from Charlie, the idea that, as shown in this cartoon, as cancers change from diagnosis through treatment and through if treatment is unsuccessful in metastasis, malignant cells change by virtue of mutation and induction of different transcriptional states as a result of a milieu. And this kind of dynamics is fundamental to the emergence of treatment resistance in metastasis. And so over the last 10 or 15 years, we've tried to develop a framework for taking cancers and pulling apart these processes. And I'd like to address a couple of particular questions around whether there is in fact detectable fitness in cancer cells that's underpinned by the genome with respect to common chemotherapeutic agents. And I'd like to show you some data that suggests that structural copy number genotypes may mediate some of that fitness. So to contemplate this, we have to recognize that we need a way of thinking about the cellular structure of cancers and where the vectorial information comes from that encodes cell identity and the fitness of cancers. And for the purposes of this talk, I'm just going to think about the left-hand end of this spectrum, which is where we use mutation and cell division to derive a lineage order, that is, derive the relationships between cells by virtue of heritability, because somatic variants can be tracked. And there's an analog of this in transcriptional space and epigenomics, which I won't have time to go into, but which sort of fills out this panorama. And the extension of the idea of lineage is that if you can make repeated observations of clone dynamics, you can link the genotypes that you observe that you're using to fitness. And it may not be that the genotype is directly itself causing the fitness, but it may be linked to other factors which cause the fitness. And so this idea has its basis, in fact, in very early population genetics that was developed by Sewell Wright, mathematical methods that were developed by Wright and Fisher. and many others in the branching process literature. And so we've set out to use this methodology to try and understand drug resistance. Now, it's not, of course, possible to make many time series measurements in humans because the process of biopsying is onerous and difficult. And so we switched to a model system where we've been able to take human breast cancers and grow them as polyclonal tumors in immunodeficient mice. And we've shown over time that these tumors do capture some of the clonal diversity. This PET-CT image is in fact of a mouse bearing a human breast cancer, which shows all of the same phenotypic properties of the originating cancer. And some years ago now, we managed to, using point mutation sequencing, demonstrate that if you put these tumors into mice and then propagate them over many months, which are sort of realistic intervals for the evolution of these cancers, that we can measure both the clone dynamics, and we can show that in a proportion of these cancers, the clone dynamics are reproducible. In other words, if you do the same kind of transplant experiment, you will get the same kind of clonal behavior, indicating that the clone dynamics are in some sense deterministic with respect to genotype. Now, the issue with looking at copy number specifically is that bulk sequencing and point mutations don't really give us this readout. And so over the last few years, we've also been developing methods for sequencing the genomes of individual cells in order to look at structural copy number changes. And one of these methods has now made its way into the literature. And just to summarize it briefly here, the idea is that we take a heterogeneous cell population, and this could be from a human tumor or a patient-derived xenograft, in fact, any model system, and we take the cells, dissociate them into single cells or single nuclei to look at the genomes, and then using a shallow sequencing approach together with the ability to array indexes, we're able to sequence at single cell level the copy number structure of cells, and then using the common copy number structure of clones, group shallow sequence cells together in order to get the depth of sequence to look at single nucleotide variation. And so what that lets us do is clonal lineage reconstruction over the entire population of cells that you might find in a cancer. And so we very recently, this just came out recently, so I'll just try and summarize this. We used this approach together with the patient-derived xenograft time series to try and identify where clonal resistance to common chemotherapeutic agent platinum, which is used in triple-negative breast cancers, might arise from. And the design here was to take time series measurements, so retransplants of tumors that were observed over many months to years, and then sample these tumors, dissociate them to single-cell resolution, and look at the copy number clonal structure using the method I described. So sequencing the genome, and then using tree reconstructions, phylogenetics, to identify the structure of the clones. And then we used a modified mathematical model that comes from the Wright-Fisher area of population genetics to define what the fitness of the clones was and ask how that fitness changed under different circumstances. So the first thing to say is that, again, when we look at this, we observe that there are reproducible, genotypically reproducible fitness measurements. That is to say, if you take an ordinary set of cells, here this was done with an isogenic p53 knockout cell line, which was passaged over a long period of time, you can show that the same clones undergo the same kind of reproducible behavior once they've been identified, up to the point where if you take downstream populations, remix them, and then sort of restart the experiment, the remixing shows that the clonal fitness determinations are accurate. And this turns out to be true also for untreated patient-derived xenografts. The heat map here shows the copy number clonal structure, which, as you can see in these tumors, is complex and would not easily be resolved by simply taking these tumors and putting them into bulk sequencing, which is how the problem has been addressed in the past. And again, we can measure the fitness and see the evolution that takes place. And again, this fitness in an untreated setting is quasi reproducible. If you take later populations and remix them at different population proportions, then restart the transplants, you observe the same kind of clonal dynamics, implying that the clone fitness that's being measured by these copy number measurements is somehow deterministic. Now, we've then taken this series and, having applied platinum treatments all along the way to induce serial resistance, ask what happens to the fitness of the clones. And what we observed in this experiment is that from the tree mapping to the clones is that there's an inversion of fitness. That is to say, clones that early on are fitter under sort of neutral conditions with no treatment become unfit when platinum arrives, and there are platinum resistant clones which emerge and dominate the population. And so this shows that you can identify these clones and implies that there is a basis for platinum resistance in copy number structure. But what's even more remarkable is that in another extension to the experiments, we then asked what happens if you take the drug away and sort of look for reversibility in the landscape. And what was observed here is that when you take away platinum and then look at clonal structure, in the early stages of platinum resistance, the clones that are platinum resistant under platinum treatment actually have a fitness cost. That is to say, when there is a competition still possible between sensitive and resistant clones, if you take the treatment away, the sensitive clones win out again and come back into the population. And it says that the resistance will therefore be revertible up to the point that all sensitive clones are eradicated. And that may seem obvious, but what the experiment defines is the sort of the measurement of that with mathematical precision. And so it implies that platinum resistance does indeed have a fitness cost. So that's what I've tried to convey here in the second part, is that in addition to learning about the genomic basis of variation and its therapeutic targeting across patients, we're also beginning to learn that copy number structural space in unstable cancers does have importance and probably has a very pervasive effect on transcription, which we are now beginning to study the effects on single cell transcriptomes and epigenomes that are underpinned by this kind of genomic instability. I'd like to stop there and just acknowledge the many workers, both present and current and past, from the lab together with many collaborating PIs, particularly my long-term colleague in the computational biology of genomes, Saurabh Shah, Alex Bouchard-Cote, and all of the clinicians who worked with us on the trial basis, as well as support from Cancer Research UK, CIHR, Stand Up to Cancer, BC Cancer Foundation, and many others who've supported our work. I'll stop there.
Hello, everyone. My name is Lillian Siu. I'm a medical oncologist at the Princess Margaret Cancer Center in Toronto. So without further ado, I'm going to introduce our next speaker, who is Dr. Aviv Rajiv from Genentech. Dr. Rajiv is the head of research and drug development from Genentech in San Francisco. She will be talking to us about cycling cancer persister cells, lineage relationships, and non-genetic mechanisms. Dr. Rajiv, please go ahead.
Hopefully you can hear me. I cannot see you when I speak, so if anything comes up, just speak loud. I will hear you. I wanted to first of all congratulate Mary Claire, and I also wanted to thank Janet for inviting me to give this talk. It's a fantastic symposium, and I'm really grateful for the opportunity. So first I have to start with my disclosures. I'm a co-founder and I have equity in Celsius and Immunitas. And until July 31st, 2020, I was on the SAB of Thermo Fisher, Neogene, and Syros. From August 1st, 2020, I'm an employee of Genentech, which is part of the Roche Group. And then my lab actually has only moved to Genentech this July. And so the work that I'm showing you here today was primarily done at MIT, the Broad, and HGMI. And so single-cell genomics has shown us that cellular profiles carry a lot of information on many fundamental features of cells, their types and their states, their differentiation, other kinds of temporal processes like responses to the environment, the gene programs that they run, and even where they're physically located and how they interact with other kinds of cells. I actually think there were talks earlier already in this symposium that highlighted some of these features. And in the specific context of disease, this gives us a new kind of vocabulary for types of studies that help us answer questions like, where might the disease genes act? In what ways are the cells disrupted? Which programs change in the cells? How are the communications between the cells affected? What might be the impact of drugs? And how would resistance arise? And so this is especially the case in cancer, where we have single cell and spatial genomics approaches that can be instrumental to help us understand this very complex ecosystem of cells that includes both malignant cells as well as non-malignant cells in the tumor microenvironment. And this applies to many processes, including tumorigenesis, drug response and resistance, and metastasis. And so today I'm going to focus on two main stories on understanding these with single-cell genomics, both with an epigenetic lens and with a genetic lens. And if I have time at the very end, I have a couple of minutes on new methods. And so I'm going to start with cellular programs of resistance and with a fact that's well established. So when you treat cancer cells with targeted therapies against mutations in oncogenes, some cells are going to survive the treatment. And those cells are often called persisters. And these cells that initially persist are often not due to pre-existing mutations, but actually to unique cell states that they assume. So here you see PC9 cells, which is a lung adenocarcinoma cell line. It's EGFR muted, and we treat it with osimertinib, which is an EGFR inhibitor. And most of the cells die, but some of the cells persist. Now, even though most of the cells that persist don't divide, some of the persister cells do divide, and they give rise to colonies. Now, persisters, and especially dividing persisters or cycling persisters, as I will call them, they can be a source of resistance and relapse. But because most of the persisters don't divide, previous studies that looked at persisters didn't really distinguish the cycling ones from the non-cycling ones. And so Yara Oren, who's a postdoc in my lab, together with Joan Brugge's lab, and this was all a collaboration between our labs, set to establish a system to track and distinguish the cycling persisters from the non-cycling persisters. So she first started by tracking PC9 cells using live cell imaging. This was done in collaboration with Michael Sabar, who's a joint postdoc with Galit Lahav's lab. And they tracked the cells along 14 days of treatment with osimertinib. And when they track the live cells, we can distinguish three different kinds of lineages. First are the non-persister cells. These are the cells that die. They're about 93% of the initial population, and they lead to these profiles that start as blue when the cells are alive and then become white. Then are the persisters that survive, but they don't cycle, or they have very few cycles. So they appear as profiles that are roughly dark blue or bluish all around. And then finally, and these cells are about 6% of the starting cells. And then finally, we have the cycling persisters. Those are the ones that start in blue and become yellow at the end, and they're about 0.5% of the starting cells. Now, live imaging is wonderful, but it doesn't let us look at the molecular properties of cells when they're alive. And on the other hand, profiling, including single-cell profiling, it gives us molecular profiles, but it's destructive, and it doesn't tell us about the history of the cells. And so in order to get both views, Yara developed a new vector that we called watermelon to let us track three features by combining FACS and single-cell RNA-seq. So first, watermelon has these high-complexity molecular barcodes, so we can track cell clones from the initial culture. Second, watermelon has a DOX-inducible M-cherry and a constitutively nuclear M-neon. And so when we pulse and then withdraw the DOX, cherry is going to onset, but then it will gradually dilute in those cells that divide, so that we can use FACS at the end in order to distinguish the cycling from the non-cycling cells. And then finally, this vector is compatible with single-cell RNA-seq so that we can get the current gene expression profiles of the cells. And so first we wanted to see if the ability to cycle was genetic or not. So we treated watermelon PC9 cells with osimertinib again for 14 days. Then we sorted the cycling and the non-cycling persisters separately, and we propagated them again without drug, and then we challenged them again with drug. And we saw that the cells that are derived from both the cycling or the non-cycling persisters, they reacquire drug sensitivity and had similar doubling times. So this means that the mechanism that allows the cells to cycle under drug is probably reversible and not genetic. So then we did a time course experiment again, but now coupled to single-cell RNA-seq. We profiled the cells before the treatment and then also at 3 time points afterwards, including at day 14. And at day 14, we actually sort the cells to 3 bins based on their cherry levels. So non-cycling cells will be red, cycling cells are in coral, and the high cycling cells are in pink because they diluted the M-cherry the most. And then we profiled each of these subsets by single-cell RNA-seq. And this allows us to examine each cell and clone by 3 kinds of information. From the single-cell RNA-seq, we can determine for each cell the phase in the cell cycle. And then from the barcode, we can look at each clone's size at each time point, and we can determine what was the majority fate of that clone, of that barcode. Were these cells eliminated after the treatment, or are these non-cycling persisters, moderately cycling persisters, or high cycling persisters, which we get from the watermelon vector at the endpoint. And then finally, from the gene expression profile, we can relate the cells to each other. This is now in a low dimensionality embedding of the profiles. And so we can relate them to each other based on their gene expression profiles. And I want to note to you that once we account for the sampling bias, some of the cells from most of the starting lineages were retained throughout the experiment. So I don't have time to go into the statistics on this on any other point that I made, but it's actually pretty nice statistics that we developed for this problem specifically, and this is all published work, so you can go and assess it there. And so putting these pieces of information together, we can now see that the cycling and the non-cycling persisters have distinct expression states, such that some of the cycling persisters, which are in light pink, by day 14, they're starting to resemble the day 0 pre-treated cells, like coming back to the original expression distribution that we saw in the beginning of the experiment. And also, most of the clones that persisted had only one fate, only cycling or only non-cycling. And the number of multi-fate lineages, where some cells are cycling and some cells are not, was actually far less than we would expect by chance. And again, there was some nice statistics for that. So this led us to hypothesize this multi-fate behavior, that cells might be leaning or maybe committed to some proliferative fate even before the treatment. And so to test this hypothesis, we did 2 replicate experiments, but starting with the same PC9 watermelon population. And then we compared the lineage size for each barcode between the 2 replicates. Because the starter population is identical, we can actually match the barcodes. And so we got a really high and very significant correlation between the replicates, and that's consistent with this idea of commitment, or at least probabilistically leaning in one or another direction. Now, you may also know that recent studies on persisters identified 2 pathways, ER signaling and ferroptosis, that affect the survival of persister cells. And that means that when you inhibit those in cellular models, there's usually fewer persisters. So we looked at each of them in our system. We treated cells with either an ER inhibitor or with a GPX4 inhibitor, which induces ferroptosis. And in each of these cases, we found a reduction in the non-cycling persisters. And as I told you, these are the vast majority of the persisters. But in the surviving population, there was a higher proportion of the cycling persisters. And this really emphasized to us that we have to distinguish the cycling from the non-cycling persisters to understand which pathways are active in each. And so that's what we did next. We specifically compared cycling to non-cycling single-cell profiles. And of course, when we do this, we first control for the cell cycle itself, because of course, that's the obvious difference. We asked what other differences besides the cell cycle. And so we first looked at programs, and we found that especially glutathione metabolism is induced in cycling persisters, but not in non-cycling persisters. And these programs were not simply upregulated before the treatment. So even though we see this commitment and leaning forward of the cells in one or another fate, these programs were not actually distinguishable at gene expression before the treatment. Then we looked for genes where the expression of the gene in the cell in a given time point is correlated with the colony size that is associated with the barcode of that cell at day 14. And the top 10 genes included 5 genes that are NRF2 targets. And NRF2 is a transcription factor that's induced by oxidative stress. So if antioxidant programs are induced specifically in the cycling persisters, maybe the cycling persisters found some way to address reactive oxygen species, or ROS. And so, again, consistent with this hypothesis, even though ROS increases in the population overall throughout the 14-day treatment, it is much higher in the non-cycling persisters than the cycling persisters, suggesting that they did find some way maybe to handle this additional pressure. And if we alleviate ROS in the population with either N-acetylcysteine, also known as NAC, or which is an ROS scavenger, or by overexpressing GPX2, the fraction of cycling persisters increases. On the other hand, when we co-treat with erastine, which inhibits glutathione synthesis, then the fraction of cycling persisters decreases. And so this all supports a model where cycling persisters manage to activate programs that address this mounting, maybe ROS pressure, and in this way they can afford to cycle, whereas the non-cycling persisters cannot. So now, of course, we wanted to understand more about the states of those cells. So we wanted to ask what might be the metabolic events in the cycling persisters that support this. And so we performed metabolomics profiling of cycling and non-cycling persisters from day 14, along with untreated cells. And each of these populations separated with a different reproducible profile, and there were 56 differentially expressed metabolites overall. And in particular, the strongest signal we noticed was in carnitine-linked fatty acids, which are substrates of mitochondrial beta oxidation, and they were significantly more abundant in cycling than in non-cycling persisters. So this suggested that mitochondrial fatty acid oxidation might contribute to the ability of cycling persisters to divide. And so we now tested this hypothesis by modulating fatty acid oxidation and checking the proportion of the cycling persisters. And when we overexpress CPT1A, which is a rate-limiting enzyme in the transfer of fatty acids to the mitochondria, the fraction of cycling persisters increased. And on the other hand, when we treated with etoxomere, which is the CPTA fatty acid oxidation inhibitor, although it has additional targets, their fraction decreased. And so this supports the model where fatty acid oxidation is somehow important for the proliferative capacity of persisters. Okay, so until now, everything I showed you tied together nicely, but it's based on a single cell line, PC9, with a particular EGFR mutation and treated with a particular targeted therapy, osimertinib. And so we wanted to ask, how general would these results be? So for this, we made a panel of 6 additional watermelon lines together with PC9. So we had 1 more lung cancer line that's driven by EGFR, 2 breast cancer lines driven by HER2, and 3 BRAF-driven lines, 2 melanomas and 1 CRC. And we treated each of them with the relevant therapy, and then we did FACS for cycling and non-cycling persisters, and then we pulled the cells together and did single-cell RNA-seq, and we could do all of the cells in one big pool because we can use their single nucleotide variation in the single-cell RNA-seq reads in order to assign them back to the cell lines. And then we focused on the 5 lines out of those that had, they included PC9, that had simply a sufficient number of cycling persisters to do decent statistics. And we found that these 5 lines shared many of the expression and metabolic features that I showed you in PC9 cells. So 4 of the 5 lines had high fatty acid metabolism program, 3 of the 5 had a high antioxidant program expression, and 3 of the 5 had a higher NRF2 program. And we also tested 2 of these lines for ROS, and the cycling persisters had lower levels of reactive oxygen species compared to the non-cycling ones. So this model generalized well. And then we also found that these features hold in vivo. So here we used an EGFR mutated mouse model treated with osimertinib, and then profiled the tissue by bulk RNA-seq at minimal residual disease. And consistent with the cell lines, MRD cells upregulated ROS and fatty acid metabolism programs. And then finally, we looked at human patient data from a very recent study from Trevor Vivona's lab, where they profiled by single-cell RNA-seq tumor samples from 30 patients that had EGFR-driven lung adenocarcinoma at 3 stages: naive tumors, residual disease during targeted therapy response, that's what we would call persistence, and then after the drug resistance was established, usually genetically, and the patients progressed. And when we compare the 3 groups, the fatty acid metabolism and ROS programs were increased after the treatment. And then we stratified each cell by their cell cycle signatures into cycling and non-cycling cells. And we saw that during persistence, the cycling persisters had a higher level of these programs than the non-cycling cells. And so the human results are aligned with our cell line and animal models. So to summarize this main part, we looked here at resistance to targeted therapy, and we focused on persister cells because most of the persisters are arrested and they don't cycle. We needed a single cell approach that allowed us to examine the cycling minority. For this, we devised this watermelon vector that allows us to track the proliferation history, the clonality, and the cell state. For individual cells, we found that we can distinguish the cycling from the non-cycling persisters. They had these different expression trajectories. There is some commitment to the proliferative state before the treatment, and then interventions in the previously described pathways like ferroptosis or estrogen signaling. They can deplete the non-cycling persisters, but they actually might enrich for the cycling ones. The cycling persisters have these different programs and this different metabolism, in particular related to reactive oxygen species and fatty acid oxidation. And we found that when we alleviate reactive oxygen species, there's a shift to more cycling persisters. And if we modulate fatty acid oxidation, we affect the persister proliferation. And these features are shared by multiple cell lines and in animal models and even in patient data. Okay. So in this main part, I focused on a gene regulation, kind of epigenetic view of the tumor under a single genetic state. But of course, the flip side of the coin is this backdrop of driver mutations that are associated with the tumors. And because of the large number of mutations for a tumor like melanoma, understanding retrospectively just by looking at tumors, not which individual mutations matter, but how combinations of mutations matter and how they cause cancer is actually challenging. So our question was whether single-cell genomics can help. And so several years ago, Eran Hodis, when he was a joint grad student with Levi Garraway in my lab, decided to engineer a system to allow us to assess this. He started with healthy primary human melanocytes, and he sequentially built 9 genetically distinct isogenic models that hit 6 common melanoma pathways, RB, MAPK, and telomerase, which together we call the core mutations, and then adding on top of them either P53, P10, or APC loss of function, and then finally adding either the P53 or the APC loss of function on top of P10. And I'm going to label the genotypes with these acronyms CBT, 3P, and A, which in all of their combinations give us all of the genotypes. We realized pretty quickly in trying to set this up that we can build these models without single-cell cloning. We rely solely on the growing, and the replicative advantage of the edited cells versus non-edited or less edited cells. And once we hit the 3 core mutations together, we get immortal melanocytes. This really only happens after telomerase activation. But now we wanted to, of course, understand the characteristics of these cells. We started by doing this in vitro with single-cell RNA-seq. And here are each of the cells' profiles. They're in color, coded by the genotype. And you can see, how the consecutive mutations are also producing an ordered transcriptional or expression progression with a couple of major transition points. For example, there's a narrow transition point when you move from having 2 mutations to having the 3 mutations in the immortal cells, what I call the 3 core mutations. Now we wanted to understand these expression changes better, so we learned gene program. One program can be active in more than one genotype. So for example, we have an interferon program that's induced by the first 2 mutations, but it's shut off thereafter. And we have an EMT program that marks this big transition through the 3 core mutations. And so on. There's a Wnt program, and a melanosome program, MIG-TORC, and phases of the cell cycle. So this was in vitro, but of course we're more interested in vivo. So we took the cells from each of the genotype and we looked at their tumor phenotype in NSG mice. So cells with the 3 core mutations together give us these small nodules in 2 to 4 months and small melanomas in 5 months or more. And when we look across all genotypes, we see how combinations of mutations are associated with distinct phenotypes. And we get primary tumor growth and at least one metastasis observed with any 4th mutation. And then we get pigmentation only with the A, the APC loss of function, or the P10 and P53 combination. And only the CBTPA genotype had this most aggressive disease with wasting. But then, of course, we pulled the tumors back from the mice and we isolated them. We dissociated them to single cells, and we profiled not just the cancer cells that we brought in, which are on the human background, but also the mouse tumor microenvironment cells from the same tumors. So first, we're looking at the cancer cells, the malignant cells. They're colored on the left again by the genotype. And you can see that there are substantial differences in the programs across the genotypes. And there's actually a nice agreement between the in vivo and the in vitro programs. But what was especially cool to us, that the in vivo programs that we saw in these tumors in the mice were consistent with the programs that you can learn from patient tumor cells from their actual tumors, even though sometimes they're called by somewhat different names. And these programs are associated with the same genotypes. So for example, both the patient-derived OXPHOS program and our in vivo mouse OXPHOS melanocyte program were enriched in tumors with mutations in the Wnt pathway and with high levels of pigmentation. And then finally, we turn to the tumor microenvironment. We analyze the programs of tumors associated, of the mouse cells that are associated with the tumors. Now, these NSG mice, so there's no BT or NK cells, but there are other cells, innate immune cells and stromal cells. And we found big distinctions in the composition of the tumor microenvironment of tumors with different genotypes and ages. The most drastic example that I'll highlight is with neutrophils, which are nearly 40% of the tumor microenvironment of cells with a CBTA and CBTP3 genotype, but they're a very small minority in any other genotype, including a very close genotype of CBTPA. And this really highlights how important it is to really assess different combinations of mutations. And then the melanocyte genotype also impacted the states, the expression states of the tumor-infiltrating mouse immune cells. So for example, M1 macrophages from CBTA and CBTPA tumors, both of which have a Wnt pathway mutation, are enriched for a TNF and an interferon program. Also in neutrophils, even though both CBTA and CBTP3 have a broad range of neutrophils and high infiltration, the neutrophils in CBTA tumors have a stronger program that's more typical for tumor infiltration. And these CBTA melanocytes express CCL2, which is actually a known neutrophil attractant. And this really emphasizes the importance of looking at other rich phenotypes. And in fact, we're exploring both H&E stains and multiplex immunofluorescence in order to better understand these features in a genetic context as well. So to summarize this second part, we built an engineered model of melanoma using relevant mutations in different combinations on the background of these primary human melanocytes. And we showed that these have distinct phenotypes. They hit immortality with the first 3 mutations, and then they accrue in aggression as more and more pathways are hit. And then single-cell RNA-seq helps us connect the mutations to changes in cell intrinsic programs, in vitro and in vivo, and they match those in the patient tumors. And also the mutations impacted the composition and the gene programs of the microenvironment. And now we can expand this approach to other mutation combinations, readouts, and other perturbations. Now, I have to admit, there is no way for me to see the audience, the timer, or anything else. So I'm not entirely sure if I'm over time, at time, or have time left. I have a couple of slides left, but I can stop here as well. You would just have to speak to me to tell me.
So next we have Dr. Daniel De Cavallo from the Princess Margaret Cancer Center and University of Toronto. And Daniel's lecture is called Cancer Epigenomics, Early Detection and Epigenetic Therapies.
So the goal of this talk is bring the cancer epigenetics to the forefront, use this for cancer as a cancer therapeutical target and also as a biomarker. These are my disclosures. So my goal here is to show this interplay between the cancer genome and epigenome. So we discussed a lot about these somatic mutations in cancer. What we need to keep in mind that many of these somatic mutations, they happens at the enzymes and the readers of the epigenetic code. So they frequently happens on enzymes that are able to read, to write, and erase this epigenetic code. And there's a big interplay between this epigenome and the cancer genome. So a lot of the mutations happens because the epigenomes allow that to happen. So have DNA methylation allow for deamination. We have mutations, the epigenetic silence of DNA repair genes that allow to more somatic mutations. And then these mutations frequently happen on the epigenetic machinery. And at the end of the day, what you have is this massive change in the epigenome. So changes in DNA methylation, changes in histone methylation, other histone modifications. And that's enable the cancer hallmark. So enable to silence tumor suppressor genes, activate oncogenic pathways, and so on. So we can use this massive reprogrammed cancer epigenome as a therapeutical target. That's the first part of my talk. And we can also use this epigenetic reprogrammed cancer epigenome as a biomarker. So we can use this for cancer early detection and for monitor therapy response. So I'll focus on that on the second part of my talk. So in terms of therapeutic application, there's multiple drugs being developed. Some are already FDA approved to target DNA methyltransferase, such as decitabine and azacitidine. We have drugs target other histone writers, such as EZH2, that can write the H3K27 trimethylation to silence genes. And there are many others in preclinical and clinical trial development. So inhibitors for LSD1, which is an erase that remove an activation mark. I'll talk a little bit about this. HDAC inhibitors and so on. And the goal of all of these epigenetic therapy is to revert this cancer epigenome and allow to reactivation of tumor suppressor genes, allow to silence these oncogenic pathways, allow to the cancer cell to to lose this dedifferentiation state and allow them to go forward within the differentiation. So that's have been the paradigm in the field for the last several years. But what we need to keep in mind when you think about these epigenetic therapies, they are actually working the whole genome. So we're not really targeting some specific tumor suppressor genes, or not really targeting differentiation pathways or oncogenic pathway. We change these epigenetic markers across the whole genome. And you need to keep in mind that these coding genes, so that's all these genes that we'll be thinking about, such as tumor suppressor, they are just a small size of the human genome, about 2% is coding. And then you have the whole size of the non-coding genome. Some part of that regulates the coding, as we discussed today. There is enhancers, there are promoters. But about half of those, they are repetitive elements. So they are endogenous retrovirus, they are heterotransposons that have been integrated in the human genome during evolution. And once you remove these epigenetic marks across these endogenous hetero elements, they themselves can have activities, and these activities can be, in part, be responsible for this anti-tumor effect that we see with epigenetic therapy. So that's what I want to focus here today. So what we found is once you remove repressive epigenetic markers, such as DNA methylation, using these genome-wide drugs, such as azacitidine or decitabine, what happens is we can start to reactivate a lot of these heterotransposons. And somehow, once they get reactivated, they can induce the formation of double-strand RNA. So now this double-strand RNA is a molecular pattern that during evolution, we learn to recognize as a pathogenous infection. So you usually have some virus that make this double-strand RNA. So the cell now can sense these, activating pattern recognition receptors such as MDA5, MAVs. This creates a signaling that activates interferon response. And this type 1 and type 3 interferon response go to the microenvironment, and they can start to create an inflammatory microenvironment. So I can activate JAK-STAT pathway, increase antigen process, increase antigen presentation. So in the end of the day, once you have these epigenetic therapies, what you do is we can target gene X or Y or Z, but we always target these repetitive elements. We always induce this formation of double-strand RNA and this antiviral response. So we call this viral mimicry. Once that's happened, we have an effect on the cancer cell itself, so the loss of cancer cell fitness, and also have an adaptive response that is adaptive immune response associated to CD8 T cell and immune response to this viral-like infected cells. So here's just one example using a colorectal cancer cell line. So that's when we first discovered this phenomenon, treated with the NAD methylating agent. And we use an antibody that's a gold standard antibody in virology to determine a cell infected by a double-strand RNA virus. And what we see here is with the decitabine row, the bottom row, we see these reds, which is the double-strand RNA being formed in these cells, even though there's no viral infection. And you can see that they are leaking to the cytoplasm, where they can be recognized by these pattern recognition receptors. Next slide, please. And for me, what's more fascinating here is the anti-tumor effect of this drug can be rescued just by removing the ability of the cell to sense double-strand RNA. So here's just a CRISPR knockout of MAVs. So now the cell do not sense this double-strand RNA, and we treat with the NAD methylating agent. So these are patient-derived cells. And we can see that by removing MAVs, the cells are lost in their ability to die or to lose a colony formation by losing the ability to sense double-strand RNA. So it could be, in this point, we could potentially be activating any tumor suppressor genes. You can induce any differentiation. The cell do not respond to the drug, even though have global demethylation. We also see this infiltrate of CD8 T cells because many of these repetitive elements, they're highly immunogenic themselves. So the endogenous retrovirus, they can be recognized by the T cell receptor. And also by induce this interferon signaling, you can have this high antigen presentation processing. So you can see here in the syngeneic colorectal cancer model, high CD8 T cell infiltration. These CD8 T cells have high interferon gamma, so they are highly active. Initially, we show with the NAD methylating drugs, we can activate endogenous hetero elements, induce double-strand RNA formation, and antiviral response. Here, we use a completely different epigenetic regulator, erase LSD1. Once we remove this eraser, the activation mark, H3K4 trimethylation, starts to build across the genome. When that's happened, we can activate gene X, Y, or Z that you're thinking about, but we always activate endogenous hetero elements. And you can see in the dot plot, this is the same antibody for double-stranded RNA. Once you remove LSD1, they start to build up double-stranded RNA. And you can see in the bottom here, in the cell lines in the culture, if you remove LSD1, the cancer cell lost cancer cell fitness. However, if you remove LSD1 and also remove MDA5, so you remove the ability of the cancer cell to sense double-stranded RNA, that we can rescue this effect. Now, we've put that in a mice model. If you remove LSD1 in the cancer cell, it's a highly anti-tumor effect. We can see the red lines, they're going down. But if you remove LSD1 and also remove MDA5, we can rescue this effect. So that cancer intrinsic effect is completely rescued by removing the ability of the cell to sense double-stranded RNA. And at the end of the day, in the final figures, in the final plot here, we see this massive infiltration of CD8 T cells if you remove LSD1 in the cancer cells. But you can rescue that by removing the MDA5 as well. So these are many examples since we published the initial paper showing different effectors, epigenetic effectors causing this viral mimicry. So we have HDAC inhibitor also causing viral mimics, many CDK inhibitors, EZH2, many mutations in the epigenetic machinery, so H3.3K7M mutation or SMARCB1 deficiency, they all cause activation for these endogenous heteroelements and activate of this viral mimic response. So these all point to maybe a physiological role to activate these heteroelements and be sensing these as a virus infection. So the concept here is we have the euchromatin, we have a heterochromatin, every cell, the cell differentiate different place in the genome will be closed and open. But if you have a situation that have complete disruption of heterochromatin because of somatic mutation in epigenetic regulators, because metabolic reprogramming or anything else, what that can cause is genomic instability and heterotransposition. That can initiate a cancer event. So the idea here is we need to have a mechanism to sense this and avoid this problem, so such a checkpoint or quality control system to avoid this. And the concept we have is these endogenous heteroelements, they play this role. They work as a sensors across the genome. Every time you start to disrupt heterochromatin globally, you activate some of those. Once you activate enough of those, you have this antiviral response, and then this cell will lose fitness and activate adaptive immune response. So what we did then is trying to identify these elements that are acting as a sensor. So what are the heteroelements across all the half of the human genome that are able to make this double-stranded RNA, they're able to mimic a molecular pattern of a viral infection and are able to activate this antiviral response. So what we did, we use a MD5 protection assay. So what we do is we get the cytoplasmic RNA, we put MD5 make a protection, and they activate once they find their agonist. And then you can treat with RNA, so you can destroy everything else, only leave the agonist of MD5. So you can actually find every element that are able to make double-stranded RNA and induce this immune response. So if you look at the donut plot on the left, you see most is blue. So these are, when you look at the total cytoplasmic RNA, most are non-repeats. But then if you look at the MD5 protected, you see the second donut plot, most is red. So these are signs, specifically ALUs, that are able to activate MD5 in this baseline effect. Then if you look at this final donut plot in the right, you see that pretty much everything is the sign. So everything is this ALU. So when you treat with an demethylating agent, all the agonists of MD5, they're coming from the sign elements, and not any sign elements. So these are two ALUs that are in an inverted position and close to each other. So they are within this window of 3 kb from each other. So that suggests that once transcription happens and go through the both ALUs, they now become complementary and then can make a double-stranded RNA. So they can make a hairpin, which is big enough to activate MD5. So these are all the sensors in the human genome that we actually perhaps maintain them during evolution and use this to sense loss of heterochromatin. Yeah, so indeed, if you look at these inverted ALUs, where they are located in the human genome, they're mainly in intronic and intergenic regions, and they're mainly downstream to orphan CPG islands. So for you not familiar, the human genome have been depleted of CPG during evolution because of deamination, because of DNA methylation. However, have regions in the genome that you maintain the expected amount of CPG, we call that CPG islands. Most of them are regulating genes, so they're in promoter region of genes, but have a lot of them that we call orphan CPG islands that happen in intronic and happen in intergenic regions, and we don't understand the function for that. But they seem to be selected during evolution to maintain the CPG frequency. So now these orphan CPG islands seem to be mainly located upstream to these inverted allos, so suggesting this whole module have been preserved during evolution to activate this antiviral response. So the model is that this is an intronic example. We also have intergenic examples that have the orphan CPG island that once we lost DNA methylation, lost heterochromatin, the build activation mark, induce transcription over the two inverted allos and make this double-stranded RNA and activate this immune response. So just to conclude this the therapeutical side, we have several epigenetic and non-epigenetic therapies that seem to activate these hetero elements, induce double-stranded RNA formation and activate of this interferon response. The loss of cancer cell fitness and adaptive immune response. This also seems to be a liability in many mutations in the epigenetic machinery, such as SMARCB1, H3.3, H7M. Also a liability in tumors with metabolic reprogramming that is decreased, so the epigenetic repression is decreased, so it's much easier to activate this pathway. This creates a vulnerability that CD8 T cells now can recognize the tumor and can infiltrate the tumor. And the major source seems to be these inverted allos located in orphan CPG islands, suggesting a quality control system, a checkpoint for broad of disrupting heterochromatin. So the next 5 minutes, I'll just discuss a little bit about the potential of using this epigenetic reprogramming, not only for therapeutic, but as a biomarker discovery. So as we discussed, there's a huge amount of change in the DNA methylation. So there's change in many different cancer types. You can see thousands of CPG sites across the genome that become hypermethylated. So they give information for us about cancer versus non-cancer, and also they give information about tissue of origin. Breast cancer methylation profile is different from ovarian cancer, which is different from a colorectal cancer. So if you can capture this in the blood, we can use this as a very interesting biomarker. So the idea here is using this for liquid biopsy discovery. So every time a cancer cell dies, they release this small piece of DNA in the blood. We can capture those. We can look for the methylation profile, and you can tell if this is a cancer-derived DNA or not. And you can tell if it's a colorectal cancer-derived DNA or pancreatic cancer-derived DNA. And the problem with liquid biopsy is the needle in the haystack. There's very few tumor-derived DNA, a lot of normal cell-free DNA derived from the hematopoietic system. Within the cancer cell, most of the DNA looks exactly like the normal cell-free DNA sequence-wise, but with the DNA methylation, we have thousands of those markers we can use to identify those. So that create a potential to have a high sensitivity with tissue of origin information that we can potentially use for early detection. So what we did, we developed an approach to capture the methylated DNA, separate that from all the total unmethylated DNA, and then we only sequence the methylated fraction of the cell-free DNA. So that's caused us to enrich it for CPG islands and allow us to identify CPG islands on shore, so identify these highly informative regions in the genome that can allow us to tell if it's a cancer DNA or not, and allow us to tell the tissue of origin. So here is using patient-derived DNA's 388 plasma samples. We can identify features of this methylome that can allow us to discriminate between a health controls pancreatic adenocarcinoma patient, lung cancer, breast cancer, colorectal, leukemia, kidney and bladder cancer. And if you use these features that we identify in the plasma and project them in the tumor, they can also differentiate the tumor tissue, and they can even differentiate cell lines from different cancer types. Suggests that we really capture the ctDNA that really provide us this information in the plasma to identify the tumor-derived DNA. So we can use this initial cohort to train a classifier, and then you can validate in an independent cohort here in 199 samples. So I'm showing you the classifier performance on the pancreatic adenocarcinoma. We also did for lung cancer and for leukemia, and we have the health controls. And you can see and have AUCs up to 0.93, 0.94 to identify the pancreatic versus else. And they hold steady in the late stage and early stage disease, so very high sensitivity. We use the same classifier now in an independent cohort that we published last year in Nature Medicine of kidney cancer and urothelial bladder cancer. We can see the classifier discriminate very well the kidney cancer versus control, differentiate the urothelial bladder cancer versus the kidney cancer, with AUCs up to 0.99 to identify those. And these are very difficult to identify in the circulation because they're low shedding DNA. We also use this in intracranial tumors, so we can use to classify different entities of intracranial. That's the lowest shedding of the tumor type, so one of the most challenging applications. And you can see the classifier can differentiate between these different entities of intracranial tumors when the imaging cannot. So allow the potential to non-invasively classify this disease and allow to indicate what clinical application we can use. So the final application here I want to show is just one example that we can use not only for detection and classification, but we can use to monitor therapy response without need to have a bespoke assay. So we can use a tumor-naive approach, just use the same assay without having to design profile for every single patient. And you can identify head and neck cancer patients that respond and do not respond to therapy by looking the ability to decrease ctDNA levels, only looking at the methylation profile. And you can see that can separate with the recurrence-free survival, with very good to bad recurrence-free survival, just by using this without having a bespoke assay. So that have high sensitivity and don't need to have tissue DNA. So in summary, we can use the cell-free DNA methylome. We're using this technology we call CF-MDIP-seq to allow for multi-cancer detection and classification with high specificity and high sensitivity. Also provide tissue of origin information and allowing to have the sensitive tumor-naive monitor ctDNA for clinical use. So with that, I will stop here. I'll be happy to discuss in the end. So this has been the work of many groups, many folks in my lab and collaborators across Princess Margaret and elsewhere. So thank you.
Thank you, Daniel. That was great. And thank you for trying again. Next we have up is Dr. Nara Jabato, who is Professor of Pediatrics and Human Genetics in McGill University in Montreal. Nara is going to talk to us about genetics and epigenetics of childhood brain tumors, bringing us to the pediatric side.
For the kind introduction. Thank you very much, Janet and Jim and the Gairdner for the invitation. It's an honor to be a part of this today and a part of those such amazing speakers before me and after me. So I have absolutely no disclosure except for once, I'm a clinician scientist and I really see those patients with brain tumors, and up to now I had limited things to offer, but we're starting really to see the end of the tunnel. And Daniel spoke a little bit about those epigenetic drugs, and I'm going to try and convince you why I feel so optimistic at last. So if you go, and the last decade has been like an amazing road for a lot of us, the event of next generation sequencing, single cell sequencing, as exquisitely shown by Aviv and other technologies, have really identified in multiple brain tumors that affect children, and especially the high-grade tumors, mainly, like super mainly, epigenetic drivers. Whether, we have a lot of hints now about the developmental origin, the cell of origin, the lineage of origin, but really it's those mutations that affect epigenetic drivers. And this is where epigenome intersects with the genome, as it always do, because the epigenome is what's giving our cell the music to make anything that we need to make at the time we need to make it. And unfortunately for those kids with those brain tumors, this music is wrong, because this epigenome is very dysregulated, and it's one hit, and this hit can have major consequences. So the oncohistone mutations that we identified in 2012 in collaboration with Jacek Majewski and Stefan Pfister, and that were discovered jointly also by the group of Susie Baker at St. Jude, have been kind of flagged as seminal discoveries in like nature milestones in cancer. Why is that? Because, as I hope I'm going to be showing you, and I'm using them to showcase how the epigenome is so important in the genesis of this new form of cancer, especially in the brain in children, how they are actually leading to cancer formation. I'll start by saying, what is it that we've identified? We've identified mutations that affect in the brain tumors, in the glioma that I study, histone 3 variants, any histone 3 variant that has a lysine 27 changes to methionine. And when it's on histone 3.1 or 3.2, it's going to be mainly in very young kids, 3 to 5 years of age, exclusively in the brain bonds. A little bit older, it's going to be the histone 3.3, and it's going to be also in the buns, but those are going to be older children, and the age could go from 6 to 7 up to 50 years of age. A little bit older, 7 to also 50 years of age, it's going to be histone 3.3, still in K27M, still in histone 3.3, but mainly associated with other driver, and TP53 is one of the major driver, and it's going to be in the brain thalamus. And actually, every single tumor, about 98%, if not 99%, if not 100% actually, of tumors within the brain midline that are gliomas, lowish or higher grade, infiltrative, are actually a story of a lysine 27 that is wrong. Either a lysine 27M mutation, and that's about more than 90% of cases, in any of your histone 3 variant, canonical and non-canonical, or high expression of a gene by the name of EZH interacting protein that actually mimic a lysine 27M on a histone 3 tail. So midline tumors in gliomas are characterized by lysine 27M and a problem in the lysine 27. We go now to the hemispheres, and now we have children, young adults, from the age of, I would say, 13 until the age of 35. And they have the other mutation that we identify, a glycine 34 change to an arginine and more rarely to valine, exquisitely and exclusively in histone 3.3, never in histone 3.1 and 2. Exquisitely, I'm repeating, in histone 3.3. And other people have identified IDH mutation that occur in the frontal cortex. The G34 occur in the temporoparietal cortex, and the IDH mutation occur in a slightly older patient, still below the age of 60 in gliomas. And we've identified SETD2 truncating mutation, and SETD2 is the only lysine 36 trimethyl transferase, and those are loss of function mutation, which lead us to say that while their brain midline is a story of a lysine 27, your hemisphere are a problem of a lysine 36 on a histone 3 tail. And I would go even further and say on a histone 3.3 tail. And those areas in the hemisphere and the thalamus are also associated with ATRX mutation. There is, in this slide, specific regional, but also temporal specificity, which led us actually to think from the very beginning that those mutations were exquisitely associated with development, and that pediatric and young adult gliomas are developmental defects, and the story of those two lysine on histone 3 variants. Beside this initial discovery, histone mutation have been identified in other contexts, in other tumors. The group of Adrian Flanagan and Peter Campbell in 2013 uncovered glycine 34 changes to a W and more rarely to an L, exquisitely also here in histone 3.3, and this is in the giant cell tumor of the bone. They had identified also in chondroblastoma, which is another benign tumor, but very maiming, a benign tumor of the bone, lysine 36 changes, also here to methionine. And we identified in head and neck cancers, in about 15% of HPV negative head and neck cancer, either K36M mutation or histone 3 mutation that lead to a change in the lysine 36 to methionine, showing yet another cancer that actually relies on a problem with this histone 3 tail. And we identified in 20% of undifferentiated sarcomas, lysine 36M mutation. So the message here is that histone 3.3 mutations are across the board, lysine, glycine, or 36, but the glycine 34 is exquisitely in histone 3.3, and you'll see why I'm emphasizing on this. While the lysine 27 or the lysine 36 changes to methionine occur in any of your histone 3 variant, and the mutation doesn't care, and it will have the same effect. So what is it? So if you go back, the lysine 27 gets monodiene trimethylated by the PRC2 complex. EZH1 and 2 are the metal transferases that deposit this mark. The glycine 34 doesn't get any post-translational modification. The lysine 36 gets monodiene trimethylated, not only by one enzyme, by multiple distinct enzymes. And SETD2 is the only one that can deposit trimethylation on this lysine 36, whereas the dimethylation, a super abundant mark that's understudied, is deposited by multiple enzymes. And one of the main ones is the NSD1 and 2. And there is an interplay with the histone 3.1 and 2 where they are deposited versus the histone 3.3. Just know that whenever you want to open a place or to silence heterochromatin or to put it in your telomere, you use histone 3.3. This is the non-canonical histone that's really actively replacing histone 3.1 and 2, an area where there's some action or zero action that you want to have. And the way I view it is histone 3.1 and 2 are maybe gap fillers, but then, you know, it's too restrictive and things needs to be done by that. The other thing that I want to emphasize here is that this lysine 27 methylation does not live with a lysine 36 methylation on the same histone 3 tail. So they go in area of the genome, they do not go together, and their roles are opposing, because lysine 27 trimethylation is known to recruit the PRC1 complex, the polycomb repressor complex 1, and this leads to repression. If it's on a gene, the gene will never get expressed. If it is in the intergenome will be also silenced. Whereas the lysine 36 trimethylation goes in gene body and is activating, and the lysine 36 dimethylation goes in gene and intergene accretion and is also presumed to be activating. And again, similar to Hillary Clinton and Donald Trump, they never could coexist. Those two marks will never ever coexist together on one mark. So we started to try and understand, okay, what are those mutations doing? What is it? Why is it like this? And we started with the K2M substitution because they were a little bit more frequent. And we exquisitely started with the lysine 36 methylation. And you'll see why, because it was the only mutation in the tumor where it was uncovered and didn't need any partner. And it proved to be a little bit more easy to study at the beginning than the K27M mutation. The first clue came by the beautiful work led initially by Peter Lewis, who was a postdoc in David Alice's lab, where he showed, and he did a beautiful experiment, that those mutations, even though the histone that's mutated, the histone 3 variant, is barely 10% of the total histone 3 pool, they were dominant, and they actually decreased drastically, and you could even think that it's completely disappeared, the level of methylation of the residue it occurred on. So if you had a lysine 27M, even in a histone 3.3, every histone 3 lysine 27 would be affected or potentially be affected and would decrease its level of methylation. And similarly, he did that, he showed for K36M, for K9M, and K4M. And the initial thought was that this mutation actually made the histone 3 tail so much stickier, and it was a beautiful inhibition of the methyltransferase domain of those histone methyltransferases, and it acted almost like an inhibitor to that effect. And there were a lot of hypotheses that went after that for this, but let's go back to our facts. So we joined forces with Peter, who created his own group in Wisconsin, and with Dave Alice, and we studied the lysine 36M because it occurred in chondroblastomas, and it was the only mutation. And chondroblastoma are thought to be of mesenchymal origin. It was easier to manipulate mesenchymal stem cell, and this is what Xiaolu, who was a postdoc in Dave's lab, did. He introduced this mutation, showed it was oncogenic and blocked differentiation, and through beautiful work where we associated transcriptome analysis, ChIP-seq, and a lot of cell assay, differentiation assay, we showed the following scenario. You have on the chromatin, as I told you, areas that are bases with the lysine 27 tri. This is a red dot that you see here that recruit the polycomb, and if it's on a gene, you silence it. And the intergenome, you interplay with the lysine 36 di in areas, and the lysine 27 tri, the lysine 36 di being a boundary that's very hard for the PRC2 complex to cross and deposit the histone, the K27 trimethylation, and to some level of dimethylation mark. Okay, boundaries. And actually in a wild type mesenchymal stem cell, you will have something that's becoming nimbles with some signal, and you could induce, using differentiation signals, differentiation to chondrocyte, adipocyte, and everything that you wanted. If you had a mutation in like K36M that would inhibit the enzyme that deposit the lysine 36 di on this NSD1 and NSD2, this is a scenario that happened. You wouldn't be depositing lysine 36 di, so your boundary would disappear. And what we showed exquisitely is that actually PRC2 was able to spread from its initial gene accretion and the intergene accretion way further into the intergenome and to kind of paste way more intergenic region, and as a consequences would recruit unduly to those intergenic region polycomb 1. And because polycomb 1 is not present in humongous quantities, this led to an active dilution of the polycomb complex, and it would be removed from areas that should be silenced in gene regions to area where it should not be there in the intergenic region. And this is actually what created a differentiation block, because those cells, when they had the K36M mutation, were not able to differentiate and led to sarcoma development in the case of the mouse in here. So 2 messages here. When one mark disappears, especially if it's a mark that's acting as a boundary, there is nothing void on your epigenome. Other marks, and in this instance, is loss of K36 dye led to a gain of K27 tri in the intergenome. And there are more than those marks going from another. There are the writers, the erasers, and the readers that read those post-translational modification that will be modified. And in this instance is one major reader, the polycomb repressive complex 1, that got diluted in its action and led to a blockade in differentiation and cancer formation. How about the K27M? If you look at this immunohistochemistry, it's super misleading, because in the bottom of the panel, you'll see a K27M mutant glioma, the IPG, diffuse intrinsic pontine glioma. You would think that, there's really zero, if any, trimethylation of lysine 27. The stain in brown is the lysine 27 trimethylation, and this is comparing to a wild type, and the rare nuclei that stain in brown are actually blood vessels and not tumor cells. And then when you look at the epigenetic, meaning the ChIP-seq that were performed on those cells, it's not lost. It's not like EZH2 inhibition where you really kind of completely demolish everything like equally across the genome that has to do with K27 tri and dimethylation. In the case of K27M, there were residual peaks that were there, and a lot of data and a lot of hypotheses came about. Is it PRC2 sequestration based on high affinity? Is it really only PRC2 that could get recruited to its high affinity site and mediate residual action? Was it a repulse? A lot of hypotheses done by a very, very valuable group, but most of them were done in cells that were not the original cells and outside of the isogenic complex. And I think that there's enough data now to support the hypothesis that we put forward in the field 2 years ago and still stands true and is going forward, is that what this K27M is doing is not really acting as an EZH2 inhibitor. It's true that your EZH2 is very slow at depositing the di and trimethylation mark. But what we showed exquisitely is the following. In a normal setting, in a progenitor cell, when you want to deposit the trimethylation of K27, PRC2 gets recruited to what we call the nucleation site. This is work by the Reinberg group and by other groups. And from there, from those nucleation sites, start depositing the mono, the di that would go and spread, and the tri that would spread, and the tri takes way more time. And what happens in the context of K27M is that this complex gets recruited without any problem in the progenitor to the nucleation site of this cell of origin and start depositing those marks. But it can never and will never be able to deposit enough K27ME3 to spread the mark beyond those nucleation sites. So PRC2 hits where it needs to hit the genome, but cannot spread the K27ME3 mark. And actually what we showed, and this work was mainly led by Jacek Majewski, is that it's one step down. So if you compare a mutant K27M cell line to its isogenic wild type, the mutant achieves level of K27 di similar to the K27 tri in the wild type, and barely any K27 tri. And we have similar data with EZIP, and this work was co-led with by Peter Lewis, that EZIP is doing exactly the same thing than K27M. It's really restraining the K27ME3 to the nucleation site, and they're not going away from the boundary. So how does this explain the oncogenesis mechanism? If you think of a progenitor and you are within your normal development, you really need to be nimble with your complexes. You need to recruit PRC2 to areas where, you know, you'll induce the patterning, the beautiful work by Dubourg that shows that the Hox gene that get there and it gets silenced forever, but other genes needs to be just transiently silenced because they would lead to differentiation. They would lead the cell to go to another state, and you just want to silence them transiently. And the progenitor needs to have a number of cells in order to create organs and whatever, so they need to proliferate. So there are some stemness genes that are pasted with the complex complex that really deposit the K4ME3 mark, A3K4ME3 mark, that's an activating mark, and the SETD2 that would deposit the trimethylation mark on lysine 36 and promote the expression of these. So those stemness genes really belong to the progenitor, and the silencing of these only belong to the progenitor. And as you receive your cues to differentiate, you remove repression from area, you keep it in others because you want to keep a patterning, and you silence those stemness gene because your PRC2 is able to move from area to another. And what happens when the context of K27M or EZIP, well, it's not moving. So this progenitor is going to be forever a progenitor, and it will cycle indefinitely in this cell state. And actually, this data is supported. I call it the Peter Pan syndrome, because this progenitor age to one point, it cannot go younger, but it doesn't and will not go older, will not differentiate. And data that was taken by Marilla Philbin and Mario Suva using single cell analysis pinpointed the cell of origin of K27M mutant glioma as an oligodendrocyte progenitor cell. And what this cell is doing is really perpetually cycling in its progenitor state, never, ever, ever disappearing. And we have further data that, for the sake of time, I will not be showing here. When we remove this mutation, we absolutely induce differentiation and further along astrocytic and gliogenic lineages. And actually, even though those cell lines have MYC amplification, P53 mutation, most of the time, if not all of the time, if you put them in the brain of mice as orthotopic mouse models, they will not grow if they lack the K27M. Or if they grow within a year, which is almost the lifespan of a mouse in our colonies. So I hope I'm convincing you that it's stall development in this case scenario. About the histone 3.3 G34 RNV gliomas, work we've done on the W mutation showed exquisitely that with time, slowly but surely, the same blockade of differentiation and the same actually ship shading. So when you lose one mark, you're replacing it with others, and you actually induce a blockade of differentiation. It takes more time, but you get it. But there's something even more about those tumors that I want to share with you. And this is actually the beauty of being able to associate clinical data, exome data, transcriptome data, single cell data, high-C and architecture data, because you become aware of misnomers and genetic mistakes or whatever mistakes. Carol Chen in my lab took a big cohort of 96 samples for this mutation. And what we showed, as we had shown initially, that the G34 mutation is associated with T53 and ATRX, but mostly it's associated to no end with mutation, and mainly in the extracellular domain, and this is really quite specific to brain tumors, of PDGFR alpha. And those mutations are actually super enriched at recurrence, where about 80%, if not more, of our recurrence samples had this mutation if they didn't have it originally. So being very, very conservative, 50% at the beginning and 80% at the recurrence, compared to any of the other high-grade glioma that barely have mutation. PDGFR alpha is very important, but it gets amplified or overexpressed, or there's an enhancer hijacking that was beautifully shown by Brad Bernstein in the case of IDH mutation. But the mutated form of PDGFR alpha and is quite enriched in the G34 glioma. So why am I telling you this? Well, when we looked using bulk transcriptomic, and we have done like a huge amount of effort with Michael Taylor and Claudia Kleinman to kind of have a brain atlas of the brain development, because we think those tumors are originating in the brain development. When we projected the bulk RNA-seq to our brain atlas done at the single cell where we've had our signature, the first surprise came, because the G34 did not match any glioma or any glial cells, but they match prenatal inhibitory neurons. And if you go back to development, if you have the prenatal, the interneurons originate from the ventral forebrain, whereas the excitatory neuron originate from the dorsal cortex. And within the ganglionic eminences of the ventral forebrain, you have radial glial cells that decide if they want to become an oligodendrocyte or an interneuron progenitor cell. And this decision is helped made by major transcription factors that are GSX2 and the DLXs. And once you acquire those core transcription factor, it's impossible for you to go back. And any OPC lineage is repressed. And we showed that by single cell anything, that OPCs are non-existent in the histone 3.3 G34 mutant gliomas. So why is that important? When we went back, we had the biggest surprise, and so that the PGFR alpha promoter was just downstream of what? GSX2, the major core transcription factor that I told you specify those interneurons. And actually, there was increased interactions between those, the promoter of PGFR alpha and an enhancer that during embryogenesis and fetal development drives the expression of GSX2. And this increased interaction was specific to G34 mutant glioma, was non-existent in IDH, was non-existent in wild type or the K27M mutant glioma. So we asked ourselves, does it exist at E13.5, the time where you have your interneural progenitor cells becoming specified during development? And we did this, and we saw beautifully that this increased interaction between PGFR alpha, GSX2, and this enhancer that drives the expression of GSX2 existed in ganglionic eminences, but the PGFR alpha promoter was completely silenced by the K27ME3, and we have some data to suggest that the histone 3.3 G34 mutation leads to the loss of this inhibition, and what we think is happening, that it's taking this mutation is shape-shifting the epigenome, and it's taken advantage of a specific loop that exists during development in ganglionic eminences, where the promoter of PGFR alpha is closely interacting with GSX2 and the enhancer that drives its expression. But contrary to ganglionic eminences, instead of the promoter being pasted by K27E3, it's activated by the changes in the epigenetic mark that are subsequent to the G34 mutation. So we have a target that's targetable. Kinase is always targetable, which is PGFR alpha. We know now that those tumors should be neuronal tumor and not glial tumor. They are misnomer, but doesn't change much. But for modeling and preclinical testing, this is major. And we know through CRISPR, because we CRISPR'd out the G34 mutation, and in contrast to the K27M, the cell doesn't care. So it's not the major oncogene. It's only here to set the stage. And after that, for this tumor to grow, it needs oil. It needs oil to put the fire. And the oil is actually PGFR alpha mutation and increased expression. Stall development is a common theme across childhood brain tumor. There really is a very specific window during the brain development, and it's really probably fetal and even embryonal, where a progenitor will be susceptible to the oncogenic hit. And this oncogenic hit really resembles what I showed you on K27M and a little bit on G34, in that it stops or stalls development, it subverts development, and those progenitors are kind of almost stuck in time and are proliferating and acquiring other mutations that makes them even more difficult to target. And they retain a self-renewing progenitor-like phenotype. And this is very true for the SMARCB1, that's part of the ATRTs, the rhabdoid tumors that are quite deadly, and other mutations that had been shown in leukemia, in AML, that affect the compass, this. So it's really something that has to do with a wrong interplay. During development, it's really almost like the music of time. You need your PRC2, your PRC1, your BAF complexes, and the trithorax. It's a battle for space. Where do I open the chromatin? Where do I silence it? How do I target my PRC1, and especially the canonical PRC1, to mainly silence in here? And this is what's wrong in those brain tumors. And this is why we need to understand them in their context, because epigenetic drugs exist. And what this mutation is doing on one mark is affecting other mark. And we've showed, and Daniel spoke about the viral mimicry. There are a level of viral mimicry because the loss of K27 dimethylation is associated with pervasive K27 acetylation in K27M and leads to a level of viral mimicry. So you could take advantage of the other marks that are unduly moving and potentially participating in oncogenesis, or affect those marks that are mediating undue repression. And I say that because it's mainly hones on K27ME3 recruitment, and K27ME3 could be potentially inhibited using EZH2 inhibitor if they were brain penetrant. So we need to kind of work with that. This work was done by multiple people in my lab, by the amazing collaboration that I have with the Kleinman lab, with Jasek's lab, with Michael Taylor's lab, with Guillaume Bourques, Livia Garcia, a new investigator, people at Saint Justine who provide sample. And I, for the sake of time, I didn't put my iChange slide, a lot of collaborators across the world. And of course, the funding sources. And thank you for your attention.
Thank you very much, Nada. We'll save questions for later. Next, we have the 2021 Canada Gairdner-Whiteman Award winner, Dr. Elizabeth Eisenhower, who is based at Queen's University in Kingston. She's a household name in drug development, and I am going to pass her to speak on driving innovation and partnerships in Canadian clinical trials. Elizabeth.
Thank you very much, Lillian, and thank you to the Gairdner Foundation and so many people behind the scenes working to make this symposium happen today. Hopefully everyone can see me and the slides. I am going to be speaking today following a series of truly elegant and kind of mind-blowing presentations, which really carve out how complex cancers are through space, through time, within people and between people. And it does rather make you wonder, how is it possible that we ever come up with therapeutics that work at all? But we do. And what I want to do in the next little while is speak to you about driving impact through partnerships in Canadian clinical trials. And I've subtitled this, The Final Translational Frontier. So in the next 20 minutes, I'm going to really talk to you about what clinical trials are, the principles for their success, how we partner, and the process. So clinical trials have many definitions. This is one from the World Health Organization, but basically they are any experiment or research study that prospectively assigns human participants or groups of people to one or more health-related interventions to evaluate the effect on health outcomes. And in cancer, the journey from the discovery of new potential cancer targets to possible therapeutics affecting those targets goes through many, many iterative steps in the lab before we make the leap to the final translational step to bring these into clinic, to definitively test them in patients, and finally determine whether those targets that we thought were important indeed are valid when inhibited in human cancers. So with all the work that goes on in preclinical and laboratory work for clinical trials, it is often asked, why do we actually need to do all of this clinical research when we have good animal models and we show repeatedly that certain interventions work over and over? And the fact of the matter is that while animal models can point you in the right direction, they do not reliably predict efficacy and safety in humans in every case. And I'm going to illustrate this with a bit of a story going back a few years from the Canadian Cancer Trials Group, where I spent 35 years in clinical research, where we were interested in looking at a drug, a matrix metalloproteinase inhibitor, as a mechanism for sustaining or maintaining ovarian cancer response to treatment after frontline therapy. And in this murine animal model of ovarian cancer, an orthotopic model, when the MMPI drug was given, you can see it had a pretty dramatic effect in prolonging survival compared to the animals with that dotted line going due south who received vehicle alone. So we duplicated this experiment completely in the human setting in a trial called OV12. And as you can see in that experiment, we saw minimal to no effective treatment and non-significant change in whether or not patients survived, depending on whether they got the MMPI or not. And that just serves to illustrate why it's important to translate these into robust clinical trials. In general, this is a bit of Cancer Clinical Trials 101, and I apologize to people like Lillian on the line who could give this talk 10 times over, no doubt. But in general, trials of new therapeutics focus on three main types of questions, all of which require human subjects. The first is the biology of the drug, learning about a new agent in humans for the first time, how it is handled by the body, what it does to the body, in other words, what it does in terms of adverse effects or toxicity, how it impacts its target, how it's metabolized and cleared. So the biology, the activity, and that is those trials address questions about understanding whether the new agent, given in whatever dose the biological study says is best, exhibits any evidence of a favorable clinical effect, normally in patients with advanced disease, and usually some markers such as shrinkage of tumors or biomarker changes. Furthermore, this stage of clinical investigation is one that can be used to refine defining which biomarker patient subsets are optimal for pursuing further. And the final stage, of course, is one of assessing efficacy. In those agents that make it to this point, how does their activity, when added to or substituted for standard of care therapy, improve outcomes to cancer patients that are meaningful to them, such as overall survival or the quality of life? And roughly speaking, these three types of questions map onto what are called phase 1, 2, and randomized phase 3 trials. And those distinctions, I would say, in the last decade or so have become somewhat blurred, with some efficiencies gained by seamless phase 1, 2, and phase 2, 3 studies. But nevertheless, how those trials are designed, those three key types of questions are vital in cancer drug development. So not uncommonly, over the many years I spent in this space, I would be asked, well, is there a recipe for success? If we do this right, can we guarantee success? And it's important to know that success is not simply a positive outcome in improving survival. Success can also mean that early on, before we waste very many resources or expose too many patients, we can learn that we're on the wrong track altogether and we should stop. So, is there a right way to do it that can guarantee success? Is there a recipe? And I hesitate to show this next slide. 10 years ago, when my older daughter got married, I made her wedding cake. And there was a recipe for that, that we could follow for success. And so if you want to make a cake, we can figure that out. But if what you really want to do is show that, for example, in the New England Journal, that KRAS mutations were the underlying factor that determined whether EGFR inhibitor cetuximab had efficacy in advanced colorectal cancer, or BRAF mutations, activated BRAF mutations, were vital in melanoma when given specific inhibitors, there is no specific recipe. But we can stock the odds in favor of success if we follow a few key principles, build the right partnerships, and understand the process. So I have 5 key principles I'm going to work through, and I'm going to illustrate them with examples from the Canadian Cancer Trials Group, CCTG, trial portfolio when appropriate. So this seems really obvious. You need to start your plan for a clinical trial with the right question. And it's amazing how hard that is for some people. There is a sense that you want to answer every question possible in your very first trial, and you really need to focus on what is it that we need to know about this drug at this stage of development. And that is the right question to address using the best science, the best pre-existing knowledge. And you could say, well, we actually have two questions that matter equally, but almost always there's a single question that will generate and drive the design. And here's an example from a trial we did about 15 years ago, an investigational new drug program trial 153, which was of a clusterin antisense in its first in human trial. Now, Martin Gleave, Kim Chi, and others in the Vancouver Prostate Center had been working for a long time trying to understand what is it that prostate cancer cells do or acquire that renders them resistant to hormone treatment and to chemotherapy. And one of the things they identified was that a protein called clusterin was upregulated as these resistance phenotypes developed. And an antisense oligodeoxynucleotide to clusterin, OGX011, was made, which in lab studies seemed to restore sensitivity. And this went through the hoops necessary to get Health Canada approval for the first in human trial. And our first question really was, does giving an antisense oligodeoxynucleotide to clusterin change the inducement of clusterin expression in a neoadjuvant setting in prostate cancer when men are getting hormone therapy? In other words, if we give them the treatment that induces clusterin, does giving this drug block that? And we did it in a phase 1 trial where patients in cohorts got ever higher doses. And men with newly diagnosed prostate cancer were enrolled, and along with their 4-week course of neoadjuvant hormone therapy, or NHT, they got increasing doses of the antisense to clusterin in increasing in each cohort. And what we looked at in a number of ways, this is the immunohistochemistry readout, was what happened to the clusterin expression in an IHC score, or the percent of cells scoring 0 with clusterin with higher and higher doses. And as controls, we used this historical control of men given 4 weeks of neoadjuvant hormones. As you can see, the clusterin level is quite high. And we observed that as we gave higher and higher doses of the clusterin antisense, we saw a failure of inducement of expression of the protein, which was a measure that the drug was not only doing what it was supposed to do, but it was doing it in the tissue and in the malignant cells in which it was directed. So the second principle is the right primary endpoint. And again, this seems obvious. The primary endpoint is one that should match the question hypothesis. And I have some examples to show you, and these are somewhat fictional, but sadly based on reality. I couldn't come up with any CCTG trials where we chose the wrong primary endpoint, which is, I guess, a good thing. But here's some examples of what I mean. This is a press release. University researchers have studied a new drug, Panacea, to determine if it improves cancer survival. And they report today that 30% of patients showed prolonged disease control and did not require a change in treatment for 6 months. Obviously, a disconnect between what they're reporting and what they were trying to achieve. And another one, a new radiation technique has been developed to improve palliation of women with recurrent cervical cancer. Tumor shrinkage was seen in 25% of patients, and this is heralded as a new standard of care to manage symptoms. And of course, both of those are wrong. Neither of them match up with the question that was being addressed. So now we get on to principle 3, picking the right population and study design. And this is where there is the most discussion, in my experience, in clinical trial design. And here I'm going to give you a more contemporary example, the CCTG trial BR31. And I have to confess that I was at CCTG and part of the initial design discussions for this trial. But after I left to move to the department head position, the team that's looking after it no longer includes me. So this trial was built on the observation that immune checkpoint inhibitors, PD-1 and PD-L1 inhibitors, show activity in advanced lung cancer. So it's a pretty obvious question to ask. Does moving these agents, and in this case, the PD-L1 inhibitor durvalumab, up front in a post-surgical adjuvant setting in non-small cell lung cancer reduce the chance of disease recurrence, and in other words, enhance the probability of cure. So the population selected for study in this trial was clearly patients who had surgically resectable and resected non-small cell lung cancer. They also had to have tumor available and submitted for PD-L1 expression data and other markers. The design of this study was, again, you might say pretty obvious. Once patients had completed whatever their standard curative treatment, whether that was surgery alone or surgery with adjuvant chemotherapy, they were randomized to a year of durvalumab or a year of placebo, and this was a double-blind study, so neither the patients nor the clinicians knew what the patient was receiving. And the primary endpoint of this study, if you look up on clinicaltrials.gov today, you will read it is disease-free survival, but in a subset of all randomized patients, those with PD-L1 expression in more than 25% of tumor cells, and which also did not have common activating EGFR mutations or ALK gene rearrangements. And we'll come back and talk about that in a little bit. A very large sample size is needed to address this question, 1360 patients randomized to detect about a 36% reduction in risk of recurrence in the subset that would be analyzed for the primary endpoint. And to do this took a humongous effort, 271 institutions across the world in 11 cooperative groups. Now, there is a lot said and written about novel population designs. There are many around. I'm just going to say a few words about those. The main reason people are talking about this is to enhance the efficiency of drug development and to focus more earlier on the most likely to benefit populations. And so that includes discussion around how do we define which biomarker subset to study, how to do trials that might have multiple arms, looking at one agent, but with various tumor types that share the common mutation or marker of interest, the so-called basket trial, or the sort of converse of that, multiple arm studies of multiple agents assigned to various biomarker-defined subsets in one tumor type, the so-called umbrella trial, and trials that are adaptive in their statistical approach as results emerge. Now, most of these types of studies are not the definitive efficacy randomized phase 3. They're more in that activity seeking or biology directed trials that I referred to at the beginning. The fourth principle is to maximize the return on investment. If you are going to do a trial, you have a great primary endpoint, you know how you're going to do it, you've done the right design, you've got the right population, but we shouldn't let the information that could be gathered, that could inform future research or help us to interpret what we see, we shouldn't let that anything go to waste that could be used in a prospective way. And for that reason, many, many trials, including the BR31 study that I spoke about a moment ago, have additional endpoints, whether they're secondary or exploratory. In this case, I list a few. There were actually more in the protocol. So that includes looking at the disease-free survival of the entire population and in a variety of subgroups defined by those three key markers: overall survival, quality of life, toxicity, and economic analyses, increasingly important, especially in the trials that are the forerunners of approval or use in the clinic, where understanding the cost benefit of a new therapy becomes increasingly important. And the final principle is really around safety and ethics. And it goes without saying that patients must be protected from unacceptable risks engaging in human subjects research. And there are two approaches to this that all trials have embedded. That is the approach that includes, prior to study activation, ethics approval from participating institutions and regulatory approval, which is Health Canada in this country, to review the package of materials about the drug and the proposed protocol to ensure that human subjects research is justified and it would be safe to proceed. but also during the study, monitoring the results to look at emerging safety signals. And as we know, serious adverse events appear during the study that can sometimes mean we have to adjust the design, refine the population, change the dose, and so on. But also during the study, we may learn something more about the drug we're studying or the treatment for the disease that we're studying that mandate an amendment to the protocol to ensure subject safety. So I also said that trials are about partnerships. And going back to my IND 153 example, of course, patients are a key partner in trials, not just because they voluntarily submit themselves as research subjects, but also because in our group and in many others, patients play a role in setting priorities for studies and in making decisions about which ones we do. But the other key partners, of course, are clinicians, investigators, and institutions across Canada. In this trial, lab and translational scientists were key to study the tumor sections that we produced and the pharmacology, funding agencies to support the work, pathologists to help us interpret what was happening in the tumor samples, data managers, quality control experts to ensure that the data was collected appropriately and on time, health regulators, ethics and regulatory experts, as well as statisticians to do the sometimes complex analysis. So a big circle of partners for this one trial of 20 to 25 patients. And when we look at the BR31 study, of course, all of these same partners are engaged, but in addition, we're international. So clinicians, investigators, and countries, regulators around the world, health economists to do that economic analysis, and of course the pharmaceutical industry responsible for the development of durvalumab, not only to access the drug and seek support for the study, but to whom the results of the study will go should it be favorable for submission. And the final point I want to make is really about trial process, and it's almost always iterative. We tend to have or some people tend to have a view that once it leaves the lab, it follows a path into the clinic through all the phases until the end. And in actual fact, while rarely I guess that might happen, it is much more like this, that there are back and forth things that are learned from the lab moving back and forth into the clinic. And I'll give you a couple of examples of how that played out. One is with an EGFR inhibitor in lung cancer, where at the beginning of development of EGFR inhibitors, it was thought that the key biomarker in non-small cell lung cancer was the expression of this receptor on the cell surface in sufficient cell numbers. But after the trials had started, in the lab, it was recognized that activating mutations in cell lines of EGFR, in fact, conferred a great deal more sensitivity to these inhibitors. So that fed back to the trials. Patient samples were retrieved, and indeed, we were able to identify that it's patients with activating EGFR mutations who were experiencing the greatest benefit, and that informed not only those trials, but future ones. And another example from emerging data is that BR31 example I gave you. When this was first designed back about 5 or 6 years ago, the primary outcome, disease-free survival. And all we really knew at that time was that expression of PD-L1 was likely the most important determinant of a favorable effect. And subsequently, with this drug and with the other PD-L1 inhibitors that are in the clinic, higher levels of PD-L1 cutoff were evaluated, but so too were was it noted that the presence of ALK gene rearrangements or EGFR activating mutations actually had a negative impact on whether or not these PD-L1 inhibitors would work. So that led to an amendment in the protocol so that now the primary outcome remains disease-free survival, but in the primary analysis group will be in the group most likely to show a benefit if a benefit is there, and that is in the subset with at least 25% tumor cells showing PD-L1 and ALK and EGFR negative. So to summarize, well-designed, expertly conducted clinical trials are the essential final translational step in discovery of new cancer therapeutics. These are complex undertakings that require really clear objectives, appropriate design, multiple partners, including clinical investigators, statisticians, translational scientists, as well as experts in trial conduct and analysis. And we are very fortunate to have a national group in Canada, the Canadian Cancer Trials Group, supported by the Canadian Cancer Society, as well as several large institutional and regional trials organizations with the expertise to do these studies. And I'd like to close thanking the many organizations that have over the years funded the Canadian Cancer Trials Group and the work I did there, but also to the many mentors, colleagues, trainees, and thousands of patients that have volunteered to be part of the studies we did. And with that, I will end.
Thank you, Elizabeth. Those are really important lessons. And congratulations on your incredibly deserving award. Next on the agenda, we have a roundtable discussion with all of the speakers so far, and this will be moderated by Dr. Victor Ling. Dr. Ling received his Canada Gairdner International Award in 1990. He is also the founding president and scientific director of the Terry Fox Research Institute. Vic, take it away.
Thank you, Lillian. Hello, everybody. We had an amazing series of talks, lectures today, and bookend by our awardees, Mary Claire Kling and Elizabeth Eisenhower. And in this discussion, if you have any question you want to ask, please just type them in the chat box. But at the same time, I think we're going to encourage speakers to ask each other questions as well, because what I've learned is that we often learn from other people asking great questions. So please do that. And I'm just waiting now for our panelists to come onto the screen. And thank you, everybody, for coming on and giving a great presentation today. At least what I've learned from all the talks is, and I think we all know this, is that cancer really is a disease of the genome. But what we're learning is that the genome is really, 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 really complex, complicated, very versatile, can do all kinds of things that we never imagined, let's say 50 years ago. And so this is a challenge. The second thing I've learned is that it is a disease of the cell division, and that the technology allowing us to look at single cells and the clonal evolution is amazing, and that we're going to learn a lot. And then finally, the cancer is a disease that needs to be understood at the person level, at the patient level. And I think this has been reminded by both by Nada and by Elizabeth, that everything we learn in the lab may not translate to people, and we really need to test them out. So here we are, and I'm going to start off by asking the first question, but I encourage the speaker to ask each other questions as well. You just pop up your hand or just shout out, doesn't matter. We have roughly around about 20 minutes or so we don't have a whole lot of time, so ask a critical question. At the end there, I really want to ask everybody or who's willing to put their prediction on the line is the future of precision cancer medicine. So just keep that in mind. So the question I have is for Sam. Sam, elegant talk, I love it. You're talking about clonal deterministic clonal evolution being deterministic. And that gives me hope, because I always thought that, these things may not be deterministic. And can you maybe just touch a little bit on, where that determinism part of it could eventually translate into some kind of prevention or therapy, or how would that work in your mind?
Yeah, that's a huge topic, Vic.
Well, you only have about 3 minutes, Sam.
Yeah. Well, I think the first thing to say is that whilst we are learning that there's fixed somatic variation in the genome that you can associate with drug resistance and what have you, that's only part of the story, and that as we've heard from other speakers and in other talks, that the way that the epigenome gets patterned and the way that transcriptome networks and protein networks change also influences. And decoding this is becoming possible, but the complexity is certainly high. I think we may soon be able to move to a position where, by making enough measurements, particularly measurements that are made in patients over time, that we begin to sort of distill down what are the key regulators in each of these domains, and the predictions of how we might then intervene might become formulated mathematically. Because if we have measurements and we can associate the right measurements, then we can intervene. The ways in which we might intervene may start to be surprising, and this involves obviously co-modulation of the immune system, for example, as well as intervening in the genome.
Right. And very clear. I think you have a burning question you want to ask.
I did. I put this in the chat so as to get your attention. I wanted to ask Daniel a question, both for researching cancer and for research in so many different traits. It's so important to have access to cell-free DNA that comes from otherwise inaccessible tissues. Could you comment, please, on the half-life of individual molecules of cell-free DNA in the blood? Does it vary depending on the source, that is the tissue of origin? Does it vary depending on whether they're methylated or not? Does it vary depending on their size? What are the determinants? And what can we do in order to increase the life of these molecules in blood so that we can work with them?
Yeah, that's a very good question, and we don't have all the answers yet. I think the field believes that usually it's a very short half-life.
Right.
Usually it's a snapshot of what's happened at that immediate time. So it's come a lot from the maternal fetal DNA that people have been used to monitor, and you can clear the fetal DNA going away very quickly after the delivery of the baby.
By short, you mean like an hour, right? I mean, truly short. Yeah. Yeah,
truly short. The methylation itself doesn't change the half-life. Will play but the methylation is very stable. So as long as the DNA is there, the methylation will be there, since it's a covalent modification. And a lot of the representation of the genome that we have in the cell-free DNA, that depends on the copy number variation, depends on nucleosome accessibility. So if you are in between the nucleosomes, they can be digested depending on the position. So all of these, in the end of the day, will influence the molecular pattern we get in the final result.
Beautiful, just beautiful. Thank you.
Thank you.
So I have a question for Aviv. The non-cycling cells, I mean, you point out that they could actually play a very, very important role. But I guess in my mind, if they're not cycling, why worry about them?
Yeah, well, there's an obvious reason why to worry about them, which is that they can transition to become cycling. So in reality, the reason we focused on the cycling ones is that because almost all of the previous studies looked at all cells together in the bulk, and given that the non-cycling ones are greater than 80% of the cells at least, it actually means that anything that was learned was really learned about them. And that's really what we found at the end. We suspected that would be the case. There was also the possibility that cycling and non-cycling would basically be the same. There were kind of two models coming up front, that the cells could be in a mostly non-cycling state and once in a while would transition to a cycling state, would do a cell cycle, will come back to non-cycling. That's one model, and in which case looking at the bulk is fine. And the alternative model is that cells actually prefer to be in one of these states over the other for a prolonged period of time, in which case being cycling or non-cycling really matters because it's part of your identity. And that's what came out from our data. We didn't actually know which one of these would be the real result. We set up a system that allowed us to ask it cleanly. And once we knew that there was this different and that cells have a fate, at least for a period of time that they tend to stick to, it became very meaningful what might distinguish them one from the other. And so even so, there's probably a transition probability between those states. And when you talk about precision medicine, I think for things that are more dynamic, which can be the state of the microenvironment of a tumor, or the state of the cancer cells themselves under a treatment and so on, that's where precision medicine starts getting sometimes a little bit into trouble. Because the genotype, it's not that it's fixed. I mean, we had Sam's talk and others to see all of the ways in which it changes, but its timescales are different. And things that can happen on a rapid or relatively rapid timescale, and that we only get usually from a patient a particular sample in a particular moment in time, which is when it's biopsies and not resections, it's also only one particular portion of the tumor. The ability to think in a dynamic way out of a more static type of data collection is, I think, one of the interesting challenges for us to the future. First of all, what can we do methodologically to let us look at things more dynamically, but also what can we take from one snapshot and infer more about things like, say, the transition probabilities between different states of cells. And I think that's actually already proven to be a very fruitful way of thinking, but it moves us from thinking about precision medicine as a point estimate, this is kind of the status, to thinking about it as a distribution and as transitions between distributions. And that's true for the cancer cells. And even though that was not a major focus of this particular talk that I gave, it's actually just as true and even more true for the cells that are not the malignant cells of the tumor, for the microenvironment of the tumor. So when you think about the status of the immune cells, the status of the stroma, the neurons that are innervating, everything is an extremely dynamic state, constantly shifts and shapes, and we need to find better ways of characterizing those distributions because that's what we really want to target in patients.
So I think, Charlie, you have some thoughts on that, because I think in your non-small cell lung cancer tumors, I believe you're probably looking at mostly the rapidly cycling cells, or am I wrong?
Well, I mean, Aviv's work focuses on non-small cell lung cancer, obviously EGFR mutant. I mean, it's a minority group of patients, but it's still a very important group. I think it's fair to say proliferation statuses of squamous tumors are higher in general than EGFR mutant disease. But, I think this issue of persister cells is extremely important. It is a problem we see in solid tumor oncology. But I wonder if I could possibly ask, I've got a itching question I'm dying to ask Mary Claire, a clinical one, and a sort of, a translational question for Sam, if I may. Would that be possible?
Yes, definitely.
So Mary Claire, you gave an absolutely amazing talk. I really enjoyed it. It's a talk I've been, dying to see for some time, quite honestly, because clinically we come across these patients all the time with very strong family histories, with breast and ovarian cancer diagnosis under the age of 50, with family members who've all had similar diagnoses, and they've had in vitro tests, germline tests, and they come out negative for all the HR genes, BRCA, PALB2, BRCA1, BRCA2, you name it, RAD51, etc. How do you advise a patient like that, where obviously in a standard, let's say, national health care setting in the UK, we don't have access to the wonderful technologies you've been developing? How do you advise a patient? What do you say? Because for me, it seems obvious they must have a germline syndrome somehow, somewhere, and we've just missed it by panel-based analysis. Right. How do you advise them?
Right. In two ways. First, I advise them to get it into a really good research unit so that there's a chance of finding them, Will Folks being an obvious one, my group being an obvious one. Yeah. But other than that, we are back to where we were in the 1960s, 70s, and 80s, and we advise on the basis of family history. I agree with you that these families almost invariably do have an underlying germline lesion, and one by one we are finding them. Of course, it's possible that a very large number of breast or ovarian cancers could co-occur by chance, but we are way, way, way beyond the tail of that distribution just by chance. So we are thrown back into, may I say it, medicine. Yeah. And that's what we do. Yeah. And make one comment and then I will cede the floor to Sam. This has been an extraordinary day. I think for me, the most gratifying part of being a Gairdner Award winner this year has been to be able to hear what my Canadian colleagues are up to. The richness of Canadian cancer research is just extraordinary. I mean, I knew it was good individually, molecule by molecule, but to hear it all in one place at one time is just terrific. Many thanks to Janet for putting this symposium together, and I have just had a fabulous time. Thank you.
Wonderful, Mary Claire. Thank you. Sam, just very quickly, a question for you. Your wonderful data on the platinum sensitivity and how fitness comes at a cost, and you can re-challenge and potentially cells will be sensitive again after drug withdrawal. Clinically, we don't see that. Patients who develop resistance to platinum and you re-challenge in general, particularly in ovarian cancer, certainly lung cancer, they don't re-respond to treatment. And that's in the absence of what we would call a complete response, for example, where we are eradicating all the sensitive cells. So patients will often have stable disease for a prolonged period of time with considerable burden of residual disease, but yet they will, when they progress, they are not sensitive to platinum, months down the line. And similarly with ovarian cancer and rapid progression post platinum, how do you sort of square the circle there?
Yeah, good question. So I've actually heard an alternative viewpoint on that, which is that it's not common, but it does happen that sometimes on re-challenge, platinum isn't often reused, of course, because of toxicity. But I have heard of examples of patients who were challenged sort of re-responding. However, maybe the part that I didn't emphasize there is that, of course, if you put selection on for long enough, eventually you end up in a state that's defined by the new genotype that is there are no more cells left that can compete with the non-resistant with the resistant state, and so you just don't have the possibility for that clonal competition to work. But in what it sort of suggests is that if there was early or partial dosing that was somehow interrupted, maybe early resistance would be reversible, whereas the later more continuous treatment would not.
Thanks, Sam. Thank you.
Any other questions?
Lillian, you had a question before.
Yeah.
Nada?
Lillian before and then me.
Thank you.
Okay.
Just a question for Elizabeth. Thank you for that great talk. And I wanted to get your thoughts on, the FDA sending out or creating a lot of thoughtful discussions on, for example, optimizing dose and endpoints. We have a lot of endpoints that are really clinical based, progression-free survival, overall survival. There hasn't been a lot of drugs that are based on a biological endpoint, such as, for example, a certain biomarker changing or clearance of ctDNA. Obviously, the validation of that endpoint is important, but do you foresee a future where a biological endpoint is as valid as a clinical endpoint in clinical trials?
Possibly. I mean, any new endpoint that's proposed usually has some theoretical advantage, usually because it happens proximally compared to what you would otherwise wait for, or it has a plausibility associated with it. So, for example, it isn't just good enough to have a complete response in your bone marrow. You need to actually clear out all cells that harbor the genetic abnormality for some different kinds of leukemias, for example. And there's a plausibility with that, but only sort of a prospective evaluation of whether changes in that endpoint actually match up to the same degree and whatever of the things, as I say, in the clinical space, we often go back to what matters for patients, and why they choose to take therapy isn't so that their marker will disappear. It is so that they will either get better or live longer or feel better. So I think the clinical trialists amongst us, while we can be enthusiastic about evaluating new potential surrogates or intermediate endpoints, need to anchor that to something in the end that does matter. So seeing a change in biomarker X always portends an improvement in things that might matter more to patients, if you will. And we've been through this with looking at what happens to PSA. I mean, in the olden days, right? Does PSA falling mean that you're going to live longer? Or does CA-125 recurring and early treatment mean that you're going to live longer? And we've been kind of disappointed when the randomized trials have been done. And it really, I think, creates the impetus not just to adopt new ways of monitoring people because they involve sexy new technologies, like can we get rid of ctDNA in the blood, but does finding that actually translate into a benefit in some of the things that later on are going to be of most importance to patients?
Thank you. I think Nada has a question.
Nada.
Yeah, I want to echo Marie-Claire. It's, I've been following a lot of your work, multiple of you in here, or using some of the beautiful tools that you've been developing. It's an honor to be part of this group. It's really, I'm humbled, and it's a major honor. Just before, so regarding your point about the cells that are not dividing, I think they're super important. And there's something in my mind, and this is a question to Charlie and potentially to Sam as well, is how come the lung cancer love going to the brain, and the brain is going to be hit like very fast to no end. Like really, the lung cancer patient often relapse within the brain, and they seem to be cured, if I can use the term, because you don't have longer follow-up from their primary. And the breast, even if they will get it, it's like it takes a lot of time before you go there. In there's something about, and it's not, it's the Esai Fiedler's work and other people's work about the soil and seed, but it seems that for the brain is something that's quite exquisite for lung cancer. And how can we tease, do you have any ideas why is that, and how can we tease more about it?
I mean, I don't think that's quite true, to be fair. I mean, squamous tumors, lung squamous tumors, don't often metastasize to the brain. Adeno's,
you're right about that.
You're
absolutely right about that.
Yeah,
you're absolutely right.
So I think there is a bit of a division there, and equally, HER2 positive breast cancer commonly metastasizes to the brain too. So, why is a very good question, and I'm not sure we really fully understand it in any way. I think there are certain driver mutations that people like Priscilla Brasilianos and colleagues at MIT have postulated are associated with propensity for brain metastases. Obviously HER2 being one of them. We've not found any similar canonical somatic mutational drivers, Nada, that would portend metastatic dissemination to the brain, at least at the mutational level. There may be copy number events that we're missing, or indeed, pertinent to your talk, epigenetic modifications we haven't yet seen. And then, of course, you've got, very elegant work around sort of neuronal tumor communications that we don't fully understand, as well as, activation of the CGAS STING pathway from people like Sam Bakum and others. So, I think we're only just beginning to understand the metastatic process, and also whether or not they're actually drivers of metastatic dissemination at all. Is this all just a stochastic consequence of tumor burden? I mean, I don't think the field has fully addressed the answer to that question yet. So,
but you're addressing it in a way. I'm sorry to interrupt. This is one of my bad habits. Is when you say that squamous lung don't go there, and adenocarcinoma, there is something that makes one go more, and there's some, and again, one of the things, ovarian cancer, but again, they don't survive. But when you shunt this ascites to the lung, this is how I learned how metastasis is kind of very peculiar.
Yeah, no, I mean, I think you're making an excellent point. I mean, in the past, I remember when I first started breast medical oncology back in the early 2000s. I'm sure Marie-Claire will testify to this, but HER2 positive breast cancer didn't often metastasize to the brain. And it was only on the introduction of drugs like Herceptin.
Exactly. This is how I was going to suggest that.
The biology of the disease changed. And I think if we could extend the lives of patients with ovarian cancer potentially, and manage
Yes,
the
and I
transmural disease
agree
and
with that completely.
we would end up with CNS metastases
This
too.
is the
So we
component.
do see that with ovary. If you live long enough, many patients who never have it early get it late in their brain. It's unusual, but it's seen. It's almost as if, yes, there are biological factors, but it's only if you live long enough in the rest of your system for your brain mets to have a chance to grow. And I remember seeing that when we first started to do trials with docetaxel, dramatic complete responses systemically, and patient after patient relapsing with brain mets while their systemic disease was otherwise under control. So there's there's a lot of biology, there's a lot of chance, but there's also how well you're controlling the disease burden from the neck downwards that gives an opportunity for things to take the time to be successful in growing in the brain.
Thanks, Elizabeth.
I'm sorry to interrupt, but we're just getting warmed up, and now we have to stop. And very unfortunately, but I just want to thank all the speakers today, and the chairs, and Lillian and Jim, and of course, I want to thank Janet and the Gairdner Foundation for bringing this group together. It's an amazing, really amazing.